There we go. Okay, this is meant to be a casual intro to the day where we just kind of talk about DDX3X, what it is, what it does inside the body, and some basics about biology and DNA that you all may or may not be familiar with. So I'll start by introducing myself. My name's Stephen Floor, and I'm a DDX3X researcher. I started I started working on DDX3 in actually 2011 as a basic scientist. I'm a PhD scientist trying to understand how cells work and how the information in DNA works together inside of cells. And it was clear even in 2010 that DDX3 was a very important part of our cells and part of our bodies. And so I decided to do postdoctoral research focused on how DDX3 works and what its structure looks like and how it's connected to the way that we all work. During that time, there was a lot of research that came out that identified mutations in DDX3X that are causative of a bunch of different diseases, actually, including different kinds of cancer and DDX3X disorder or DDX3X neurodevelopmental syndrome. And so I continued working on it as a faculty member at UCSF for about 7 years. I've left UCSF in 2024 to join biotech, where I've both focused on RNA therapeutics and I'm trying to start a gene editing company right now. So I've experienced both in academic research and in biotech and in many different kinds of rare disease. And right now I'm a science advisor for the DDX3X Foundation. So I'm not actively doing DDX3X research, but I'm still connected with the community. And I'm here to moderate and try to connect the dots for you all today. This session is really for you all. This is about connection. It's about learning information. It's about answering the questions that you all may have. So I don't want to just stand up here and talk. And please, when we have the sessions later with group panels and stuff, ask the questions that you want to ask. If it's to the panel, ask it to the panel. If you want to ask a question that comes to your mind, to the room, ask it to the room. Like, let's really try to make today a dynamic day where we can all work together and help each other. So I'm going to start off with just some really, basics that some of you may be familiar with, but I think it's really important that we set the foundation. So, all the different cells in our body, all the different tissues in our body are made up of cells, and all those different cells are very specialized and have different functions, but they have mostly the same DNA, right? So the cells that are in my heart look very different than the cells that are in my brain, even though they have basically the same DNA. And it's been known for a long time, since the 1980s at least, that DNA changes can lead to different forms of genetic disease. So the first marker that really connected the dots was in Huntington's disease, where people figured out that DNA changes are actually driving that disease. There's now many, many classes of genetic diseases that are known to be caused by sometimes small and sometimes large changes to DNA. And there's many ways that a change to a DNA that's in all the cells of your body can affect just part of your body. And that's something that we could get into if you want to. So when it comes to DDX3X, it's located on the X chromosome. So most girls have 2 copies of an X, most boys have 1 copy of an X. But when we look at where the material inside of the DDX3X gene is expressed, we find it in basically all the cells of the body. Okay, so nearly every tissue that you look in has cells inside of it that are making the DDX3X protein. And that's one of the reasons that it's so important. That's one of the reasons that you see in folks that are affected by DDX3X impacts in many different organ systems in the body. It's because this is a gene that really is important for a lot of the things that we do. So in around 2014, 15 was when the scientific research started coming out. There had been people diagnosed with DDX3X before then, but the research came out around that time that really identified that DDX3X is actually a much more common cause of intellectual disability and developmental delay than we'd previously recognized. And one of the studies that identified this came out in 2014, where they took about 1,100 people that were affected by undiagnosed developmental disorders, and they sequenced their DNA. So we'll talk about how you can do that later on in the talk. And what they found was all these different dots here. It doesn't really matter exactly what they are. The red ones were genes that were already known to be causative of developmental disorders, and the black ones were new. And what I think is interesting about this is that they drew these red lines. So this is where they were going to cut off, genes that are important and genes that are not important. And DDX3X is right there, sitting right in the corner, right where if it had been, just a couple people that hadn't had it, it probably would have been missed in this panel. Which is not to say it still wouldn't have been found, but serendipity. Shortly after that, there was another research publication that came out that mapped a lot more information about what's actually happening in folks affected by DDX3X. And it was found, here you start to see a little bit more information about some of the things that you may have heard about. So what they found is that 19 of the 35 people that they studied here have loss of function alleles, which means a change to the DNA that prevents proper functioning of DDX3X. And then you're hearing things that you may have heard about, again, nonsense mutations, splice site mutations, exon skipping. We'll talk about what some of that means. And then they found also the other variants or mutations were so-called missense variants. So this means that you still are making the protein, but there's just one piece of the protein that's been changed. And so they're starting to map where those are happening. And these green parts are pieces of DDX3 that are especially important. And you see that the changes are happening in the parts of DDX3 that are really important for it doing its function. They also included in this, what many of us are familiar with, right, which is what the community of DDX3X people sort of look like. One of the things that I think is fun at this meeting is that you'll see a lot of facial similarity and behavior similarity in people that have DDX3X, and so they started to describe, what folks sort of on average how they develop and how they appear. So how do they actually do science like this? So all of the cells of our body have DNA, right? And so we can take, for example, blood, which also has cells in it, also has DNA, and then we can sequence the DNA that's in that blood and compare it to the known DNA on average that most of us have. All of us have small genetic differences, right? Most people on average actually have millions of genetic differences between each other, but most of that happens in parts of our DNA that's not making a protein. It's not involved in the actual functioning of our body. It's just kind of variation that's drifting around. When we have a change in something like DDX3X that is in a gene, it's in a piece of our DNA that is expressed, and it's making a really important protein that's in all the cells of our body, like we talked about. So the first way that we can identify this is the most thorough, it's the most expensive. It's being more and more commonly used because one of the things that's happened since around the 2010s to now is that the cost of sequencing has gone way down. Actually, just last week was a record for the fastest human genome sequenced. It was sequenced in about 4 hours. So you can now imagine, going to the emergency room and having your whole genome sequenced and having the doctor come back and saying, this is the DNA change that you have right now, which is in 2010 was absolutely unbelievable. I still think it's kind of unbelievable. So with whole genome sequencing, what you do is you take all the DNA inside of some cells, you break it up into these tiny pieces, and you use a sequencer to sequence all of the DNA information of all 3 billion bases that are in your cells. And you compare that on average to what we know about where most genetic variation happens, and then you find variants, right? You find something that's different. Sometimes you do that in a trio, right, where you have 2 parents and a sibling, affected child. Sometimes you do siblings, right, and you can use that to compare within a family. You can also do what's called whole exome sequencing. Some of you, again, may have heard about this. This is where instead of sequencing the entire genome, this is about 3 billion bases, now you're only sequencing the so-called exome, which are the pieces of the genome that are made into RNA that then goes on to make proteins. This is much cheaper and easier to do because a huge amount of our DNA is not functional. It's just kind of sitting around. It's in our cells, but it's not going to be responsible for a disease. And changes there are not going to be as likely to be causative. So oftentimes, people will start with whole exome sequencing because it's just much cheaper and faster. And then you can do what's called a panel or a single gene or target region sequencing, where you're now saying, I know that these 10 or 50 different genes, pieces of DNA, are important for this class of disorder. So for example, there's a lot of autism panels, and people will know these are the top 100 or top 500 most likely genes to be connected to autism-like disorders. And so I'm going to, instead of sequencing even the whole exome, I'm going to look at this person's clinical presentation and I'm going to say, I'm going to start by just sequencing these 500 genes, and I'm probably going to find something there. And if I don't, then maybe I'll move up to whole exome. And if I don't find something with whole exome, then maybe I'll move up to whole genome, right? So oftentimes testing works that way, although these days I have to say more and more we're starting with, whole exome or maybe even whole genome because the cost of sequencing is just going down so fast. Any questions about any of that? So how this actually works inside of our bodies is that we have this DNA where these DNA changes are happening. This is basic biology. It's transcribed to make something called an RNA or a messenger RNA. This is basically, I like to think of it as like the, somewhere in Lego headquarters, they have the master Lego map that has all the instructions to make some Lego kit, right? They're not going to send that in the mail to us because then they would lose that. Okay. The messenger RNA is like the Lego instruction booklet that comes in the Lego kit, right? Where it's your copy, it's your instructions, you give it to your kids, they shred it, it's destroyed, right? But it's not a big deal because there's still the master copy in, Lego HQ, right? And then the protein is actually the Lego model that you've produced using those instructions, right? It's what your kids are actually going to play with. It's the piece that's doing sort of the work inside of cells, right? Proteins are, what carry around oxygen in our blood. Proteins are what help us to think. Proteins are how our muscles contract, right? These are the pieces of our body that are really doing the work. So the mutation, that you may have mapped in DDX3X is a DNA mutation, right? That then causes a change in the mRNA that usually causes changes to how the protein is made. Sometimes it causes changes to the mRNA itself so that the mRNA is not stable and you're not making protein at all. So those are sort of the two main classes of mutations we see in DDX3X. Are these missense mutations are ones that are going to end up changing the protein. You've built the Lego not quite right. And so instead of it being a tiger, it has a, moose antler coming out of its head, right? If you have a mutation in a splice site or a frame shift, that's actually probably going to cause the mRNA itself to be destroyed. And so then you're not making enough of the protein. I'm a visual person. I like to think about how things look. So this is what messenger RNA actually looks like. It's quite big. It tends to be kind of long and spiraled around and clumped on itself. Proteins, by comparison, are relatively small. So this is a protein called myoglobin, which is involved in the functioning of your blood. And this is an actual picture, a real picture zoomed in using an electron microscope of an mRNA in the middle here. These like chunks on them are called ribosomes, which are the piece inside of our cells that actually takes the mRNA and it translates it to make protein. And then you can see these kind of strings sticking out that are actual protein molecules that are being produced off of this messenger RNA. And this is what it actually looks like inside of a cell. So if we zoom in really closely, this is a piece of a neuron. There's actually a connection here between two neurons. There's one piece up here on the top, and this piece down here on the bottom is a part of a neuron. These big blobs that are inside of here are called mitochondria. Those are what make a lot of the energy inside of our cells. And these little tiny black dots here are all ribosomes that are making protein off of mRNA. You can see they're sitting right here at the connection between two neurons that are talking to each other. And it's actually known that learning, receiving new information involves the making of new proteins. So our neurons are connected with each other. And when they're activated and we're learning something, they're making new proteins and they're making that connection stronger. And just as a brief aside, mRNA is a molecule that's made inside of our body. mRNA is also used to make mRNA therapeutics. So if any of you have had an mRNA COVID vaccine, that's basically just taking the mRNA instructions to make the COVID protein and putting those into somebody so that it can skip the DNA part. You start with the Lego instructions to make the COVID protein, you make the COVID protein, and it vaccinates you. So that's actually quite effective. Right. So, when we think about breaking down an mRNA, and I'm talking about this because DDX3 is actually related to what mRNAs do. So it's both made by an mRNA and it acts on mRNAs. We can basically take an mRNA molecule and split it up into three parts. So there's this part inside the middle that we call the coding sequence, which actually has the instructions to make the protein. And then there's these two parts on the ends that are called untranslated regions that are sort of like control knobs. So I like to think about this. My kids have a troll mixer for making music. It's a DJ mixer, right? And so they have different control knobs on there that change the volume or the delay or whatever, right? And then these are the beats that, make the protein, right? So this is how an mRNA really works, is that you have these instructions in the middle that are there to make a protein. And then you have regulation, a lot of regulation that controls where do we make the protein inside of cells? How much of it should we make? Should we make it all the time? Should we only make it sometimes? Those kind of decisions are super important. And those are the decisions that DDX3 is involved in. So I'm going to take a brief, I'm going to pause for a second. Does anybody have any questions on that? We're going to go into the science more today. So if you have questions, please feel free to jump in. When you have these DNA changes, like I mentioned, they can affect the protein, they can affect the mRNA themselves. And so you may have heard about these many different types of mutations, and I just wanted to illustrate what those are. So on the left, as an example of two mutation types, you have nonsense mutations. Nonsense means that you're making a stop codon. It's not a sense codon, so it's a nonsense codon. Anyway, so if you have, normally our mRNA is broken into pieces of 3. It reads out 3 letters at a time of the DNA that's made into RNA, and these are called codons. Okay, so each little group of 3 corresponds to 1 amino acid, which is like 1 Lego building block piece that goes into making a protein. And so normally if our RNA has a GAA, that would make this particular glue. If we change it to UAA, that instead is a stop codon. So instead of telling the ribosome to add a new piece of this protein, it says just stop totally. So if we have a stop in the wrong place, right, of course, we're not going to build the whole Lego kit. We're only going to build half of it. It's going to be a tiger with 2 legs and no body, right? So this we call a nonsense mutation. And if you see it diagrammed on like a gene DX sheet, it's going to look something like this. So this is telling you that you've changed the 150th nucleotide from a G to this should be a U, and then you've changed the glue to a stop. A missense mutation, on the other hand, means that you're changing actually which Lego piece you're adding. You're changing the piece of the protein that's being added on. And so now instead of going from a glue, you make an ASP, and that's going to look something like 152A to C and P glue 50 to ASP. Okay, so this is a missense mutation. We're still making the protein, but it has a slightly different building block. This is a nonsense mutation where you've stopped too early or not making the protein at all. And then it gets a little trickier because you can also have insertions or deletions, and these are often what lead to what you may have termed a frameshift mutation. So, all of our mRNAs have these blocks of 3 that are read out 3 at a time. The ribosome is very careful about making 3 steps at a time. If instead we now have some DNA change that's added a U, say, that's going to be written down like this. These can get very complicated. They can be really long with parentheses and stars and all kinds of stuff. If any of you want to talk about what a particular version of this means, I or other folks here would be happy to try. I don't know all the rules myself. It's like whole pages. Anyway. So if you now add a U, instead of, this was, over here we had the same number of nucleotides, we just were changing one, right? Over here we've actually added a U, so this is listed as an insertion in between 305 and 306. And what that's going to do is now we take our reading 3 at a time, right? And we shifted it by 1. So that now this, instead of being a AGU that's read as a serine, this is now a UAG, which is read as a stop. Okay, so inserting this 1 U has changed this from a piece of a protein to a stop. On the other hand, down here on the bottom, we have a different related example where we now have a deletion. So here, originally we had a GCC, now we lost the G, that's listed as 300 del G. And this also causes a frame shift, right? Because now we've lost one of the 3 pieces of the codon that we're reading at a time. But instead, in this example, now it changes this alanine to a proline. These ones are still making pieces of a protein. And what that means is that it's going to say, alanine 100 to proline, and then it's going to say frame shift with a stop somewhere over there. Okay, so it's going to make part of the protein in the way that most people are making it, and then it's going to get frame shifted, and it's going to make the rest of the protein in a slightly different way, and then somewhere it's going to stop. So when you see, a gene DX or some kind of genetic diagnostic report, it's going to have both these C terms and the P terms. That's going to talk about exactly the changes that are happening in the so-called cDNA, which is basically a representation of the mRNA, and the changes that are happening in the protein. And so depending on what changes are there, if you have a P with a stop, that's probably a nonsense mutation. If you have a P with 2 things on both sides, that's probably a missense mutation. If you have a P with a frame shift or a stop, right, that's probably a frame shift mutation. Yeah. Don't apologize. Yeah, so a duplicated T is going to be a lot like an insertion, right? So that's where you have, 2 Ts that are basically next to each other instead of 1 T. That's going to also cause a frame shift. So if you take this stop codon and you put another T in, it's not going to be a stop codon anymore. So it's going to cause now the protein to somehow keep going. Yeah. So what that actually does to the protein, maybe, maybe what you're wondering. I think there's a couple things that a change like that could do. One of them is that that extra bit that you're making could be unstable. And so it could make it so that the protein is produced and it all sticks to each other and the cell figures that out and it destroys it all. So it might look like you're not making protein off of that gene, even though it actually is able to make the protein. Another thing that it might do is that there's many ways that cells figure out when an mRNA doesn't look quite right. And one of them is if you keep translating too close to the end, the cell decides something's wrong. And so it's possible that a change like that also could cause the mRNA to not be stable. So you're not making much protein because it tries to make protein and it's like, this doesn't look quite right. And then it destroys the mRNA. But exactly all the changes that are out there and how that, like, this is, it gets kind of tricky. Mm-hmm. Yep. So there's one other major type of DNA change that I wanted to touch on, which is called a splice site mutation. So I mentioned whole genome sequencing and whole exome sequencing earlier. The genome is all of our DNA. The exome is just the parts that are making mRNAs. And it turns out that because biology, when DNA is made into RNA, there's only some parts of the RNA that are actually used, and those are stitched together in a process called splicing. So you have so-called exons and introns, and the mRNA is going to be made, let's say, from the left here to the right. And as it's being produced, there's certain nucleotide combinations that come out, and there's another whole machine inside the cell called the spliceosome that sees that, and it cuts out these introns, and it stitches together the exons, okay? So if you have a change, for example, that's outside of an exon, right? So this is not a piece of the protein, not a piece of the mRNA that has the instructions to make the protein. But let's say it's just right here in this intron, right next to the exon, and we had a G to an A substitution, right? It's gonna cause this site to not be recognized as a splice site. And so now instead of putting together exons 11, 12, and 13, we're gonna stick 11 and 13 onto each other. And this can do all kinds of different things. It can destabilize the protein like we just were talking about. It can cause a frame shift because not all exons have the same length. So this can cause a lot of different kinds of changes. And this is listed slightly differently in the way that these reports work because this nucleotide is not actually part of the mRNA. And so you'll see things like 77 plus 1G or 104 minus 1 A to G, 217 minus 2 del TCT, stuff like this, right? Where these pluses and minuses are saying that the DNA change is actually happening in an intron region that's not part of the mRNA, but it's probably involved in the instructions for how the mRNA is stitched together. So the bottom line is, there's many different DNA changes that can change how DDX3X is expressed. It can change the mRNA or how stable the mRNA is. It can change the actual protein that's produced. And all of those will result in different impacts to the cell and different manifestations in people. So why are we talking about mRNAs? So DDX3, really is a protein that itself is produced by an mRNA, but it also actually interacts with mRNAs to change how other proteins are produced. So this is an illustration I made a long time ago when I was first starting to work on DDX3 that has all the different parts of the translation machinery that are involved in, finding an mRNA and deciding I'm going to make protein off of this mRNA. DDX3 is a particular interesting kind of protein where it's basically a motor. So it can go find parts of the mRNA or other RNAs that have stuck to themselves, and it can go find those and it can unwind it. So it uses energy inside the cell. It's like a little motor that goes around and it looks for pieces of RNA where they're base paired and it can separate those. And this can be really important for helping all these translation things to find this start codon over here. In this example, if there's a really stable piece of the RNA that's folded on itself, the rest of these guys might not be able to make it over here to the start codon, and DDX3 comes in to try to help them to make it over here. And we just heard some really exciting work yesterday at the science session about how this is happening in the heart, and we have previous work on how it happens in the brain and many other types of cells. So some of the first science that described, how these processes are all connected, we have DNA changes that are causing DDX3 changes. We have some function in translation. There's other functions that DDX3 does as well. We described in a paper in 2020 with Elliot Scher, Debbie Silver, myself, and a lot of clinicians and researchers, where we studied how these different DDX3X mutations are related to clinical outcomes, to brain development, and to RNA metabolism, or this idea of taking a double-stranded RNA and separating it into single-stranded RNAs. I'm just going to tell you a little tiny bit about it. So these are some of the neurological features that you may be familiar with. This is data from Elliot Scher's. This here in the middle is a normal brain. These are MRIs of brains. And there's two different features that Elliot's group and other clinicians noted in this paper. The first one on the left here is related to this structure in the middle of the brain. This is called the corpus callosum. Our brain is split into two halves, and the corpus callosum is basically the part of our brain that helps the two halves to talk to each other. Okay, so there's this structure in the middle brain here that looks like this. You can see this individual on the left with DDX3X syndrome. These are the mutations down here at the bottom. You can see these are all missense mutations, right? Amino acid changes that are still producing DDX3 protein with a slightly different piece. This person on the left has what we would call a defect in the corpus callosum, where you can see it just doesn't look quite right, like the one in C. This person in B has so-called agenesis of the corpus callosum, so it hasn't developed really at all. And if you don't connect the two halves of the brain properly, that can lead to a lot of challenges. These two people on the right, actually, these are 4 people, have a different type of manifestation in the MRI that you may also have heard of called polymicrogyria. Poly means a lot, micro means small, and gyri are the folds of the brain. So this basically means that you have a lot of small folds of the brain. This is harder for me to see as a non-neurologist, but neurologists tell me that if you zoom in here, then you can see that the outside of the brain is very folded. And that also can cause challenges in terms of brain signaling. So, this is when I showed earlier some of the first DNA studies that had mapped DDX3X changes in people that were affected by developmental delay. This is when we really started to connect the dots on, this is what's actually happening inside of people. So we went on then to do some other studies. You'll hear a lot about animal research today. We use animal models to understand biology for a lot of reasons. One of the biggest ones is that it's very hard to grow a brain outside of an animal. It's hard to grow a brain in an animal, but when we want to study how the brain works or how it's connected to other systems, we use animals for that kind of research a lot. This is work that was done by Debbie Silver's lab. Debbie Silver is here at this meeting, so you can certainly talk to her if you are curious about brain development. This is a picture of a young mouse. This is a picture of a mouse brain, which looks a little bit different than a human brain. And normally in brains, when neurons are born as an animal is developing, they start off in this bottom area that's close to the middle, and then they differentiate into neurons and they migrate out towards the outside. So as something becomes more mature, it moves from this VZ at the bottom all the way out to the top. And so what Debbie's lab was able to show is that if we take normal developing mice and we follow neurons as they're developing, they go all the way from the bottom to the top. You can see these green dots are where neurons are, and they've migrated all the way to the outside. If we go in and we make a manipulation to the mice with a gene editing technique where we delete DDX3X, so this is not a mouse that has DDX3X syndrome, this is a mouse where we've gone in and we've intentionally gotten rid of DDX3X protein totally, we now see that basically there's no green at the top here. And what that means is that it's possible that that's going to become more common. Right now, it's still not very common to have those kind of one-off gene editing therapies. And the biggest reason is that whenever we're developing a new therapeutic, the number one thing you want is for it to be safe, right? And you don't always know with the gene editor all the things that it's going to change inside the cell. And so there's a lot of work that happens ahead of development and deployment into people of these gene editors to make sure that they're only making the DNA changes that you want. Because if you make the wrong DNA change, that can cause a whole other set of problems, right? And you don't want to go in and make things worse. It's like priority number one. So this is just a small illustration of how these work. So in gene therapy, you're adding in a piece of DNA that then has the instructions to make the missing protein. In gene editing, you're actually going into the DNA, you're changing the DNA base back, and then you're making only the correct protein. I think another challenge when it comes to thinking about gene editing, in particular with DDX3X, is that, like we talked about earlier, it's in all the tissues of your body. Okay, so if you want to actually make that DNA change back, you have to ideally do it.
But not only that, but the company.
You have to ideally do it in all the tissues of the body, which is just logistically hard if you think about material to.
Here's Devo.
Thank you. So I think the two things that, so we're going to talk about gene therapy later today. The DDX3X Foundation has funded a lot of research in gene therapy. I think in particular for folks that are affected by nonsense or splice site frameshift type variants, where again, you're missing the mRNA, you're not making the protein at all, that has the most promise. When it comes to folks affected by missense variants, that's a little bit more challenging because those may be problematic on their own, even if you have a regular copy of DDX3. So what I think we can all do today, right, is two things. The first one is symptom management, right? So you all know, your DDX3X folks, you know the things that they're dealing with. You can talk to your doctor about those things, right? If there's nausea, if there's GI issues, if there's sleep stuff, like you can talk to them about that. There's a lot of medications out there to treat individual symptoms. And I would encourage you also to participate in the DDX3X Foundation has a registry where we try to collect information like that. And that helps to get back to clinicians that then decide, oh, we're going to actually measure that more. We're going to get more information about that that may try to improve the standard of care for everybody. And the second thing that we can do is, we can teach each other and learn about it, and we can encourage, everybody affected by DDX3X to progress, right? We have adults in the room, we have adults at this meeting that are DDX3X folks, right? If you believe and you push development of your DDX3X family members, right, they're going to get to the furthest point that they can, right? Versus, my son has ADHD, he was diagnosed with ADHD. That doesn't mean he can't pay attention, right? It means that if he's bored by something, he's going to let you know about it, right? So, you don't want to treat that as a label and say this is something that somebody cannot do, right? You want to try to encourage them to do what they can. Okay. So I wanted to end with just a couple of resources. I can share these slides also to Chelsea. You can feel free to take pictures or write them down if you want to. The first one is a review article that an MD PhD student in my lab, Margaret Elliott Scher, who's a neurologist, and I wrote in 2023, where we really tried to connect the dots between all the different mutations that are out there in DDX3X in both neurodevelopmental delay and different disorders, and connect that to how the protein is working, and also connect that to what we might be able to do about it. We tried to write it in a way that it would be both scientifically language-oriented and readable by people. So, hopefully it's not just total jargon. And I wanted to just touch on this briefly for a couple reasons. First of all, because I think it's a good resource, but second, because it contains the word cancer. And so some of you may have heard about, cancers where there's also DDX3X mutations that are associated. For the most part, we think, based on data, that those are not the primary cause of cancer, that somebody has a change in a different gene, and then DDX3X is changed on top of that, right? So there's many cases in the cancer field where you have a big change that makes it much more likely that somebody develops cancer, and then you have a second change that kind of puts them over the edge and they actually get cancer. In almost every case, DDX3 is that second change. It's not a primary cause of cancer on its own. So if you've, been reading about literature and seen stuff about cancer, the bottom line is we don't see at least a large change in cancer incidence among DDX3X disorder families. Second resource is a gene review that was written by Elliot Scher and a bunch of clinicians. This is written in 2020. It basically contains, this is oriented towards clinicians, so this has more jargon than the paper that we wrote, right? But this is really about, what is this disorder? What are the symptoms? What can you do about it? What's management? What are expectations? How do you diagnose it? What else should you be looking for? How do you compare it to other disorders? This is a resource that's really written for clinicians. So if you're talking to a clinician and they're like, what are you talking about? Or how do I know it's not this thing? Or they're confused about what's going on, I would point them towards either this or this paper, which is from folks at Mount Sinai. This is a newer paper from 2023, but it's very similar. It's about, what is DDX3X syndrome? What are the different findings? How do you care for folks? How do you treat? What's the clinical management strategy for people that are affected by DDX3X? You can just go to that. You can search up on the internet, the title of it, or you can search for the authors, or you can write down that obnoxious number, or you can take a picture of it. Yeah. Yeah, I can certainly share the slides. Yeah. And you can also email me. Happy to talk about this kind of stuff, but Chelsea also has a lot of these resources. Okay, so those are the slides that I have. I'm gonna just stop and see if any of you all have any questions or other stuff you want to talk about. I think in about 5 minutes, we're gonna start the proper opening remarks for the session today. Any other questions? Okay, let's take a 5-minute break and we'll be back at around the top of the hour. Thank you.
Yeah. We're gonna call it, we're gonna do our.
Nope. Hi, everyone. We're going to get started. Nobody cares. Thank you. Good morning and welcome to the 11th Annual Scientific Conference, October 17th, and my co-chair's birthday. So let's embarrass Beth and say happy birthday. Thank you. Thank you. Of course. Next slide. How do we do the next slide? Sorry. Yeah.
Nothing says happy birthday like hosting a conference for 100 people. Our mission is to connect families, resources, and the medical community to advance research for a treatment or even better, a cure for DDX3X syndrome. So our ultimate goal has always been to accelerate brain function in individuals affected by DDX3X syndrome through advances in cell and gene therapy and pharmaceuticals.
We have tremendous gratitude for our scientific advisors, researchers, and clinicians, and we want to recognize all of them who go above and beyond to support our community, many of whom volunteer their time out of pocket to support us at our conference and in meetings throughout the year. If you are in the room, please stand to be recognized. To all of our clinicians, researchers, no one standing, and scientific advisors. Thank you. Seriously, Beth is in fashion. I'm in entertainment. We know nothing about science, so we seriously could not do it with all of you. So thank you so much.
So we've had a really exciting past year. We have expanded our natural history study. We had our 5th annual DDX3X Family Day in Arkansas, thanks to Ashley Chastain and family. We've expanded our scientific advisory board. We've expanded our patient-focused research network. And Chelsea, our executive director, along with us, we've attended some great conferences. The Chan Zuckerberg Initiative Conference, they're the people who really got us going with our Rare as One grant that we were the first cohort to receive and got us up and running. The Global Genes Patient Advocacy Summit, the Rare Disease Partnering Summit, and the NORD Innovation Summit. And then some of us also did a super exciting thing we've done a few years in a row, which is really impactful and empowering. We went to Rare Disease Week and lobbied with Congress on Capitol Hill.
Grants awarded by the foundation. So we have raised $2.4 million since inception. $600,000 was earmarked for capacity building per the terms of the funding, and the foundation has been awarded $1.2 million in research grants to date.
So just a brief bit about our history. So Liz and I started the foundation back in 2015 after the first patient in the United States was diagnosed in 2014, who was my daughter, Virginia, who's jumping for joy on that hay bale and in the room right now. Raise your hand, Virginia. Scientists estimate that DDX3X syndrome accounts for 1 to 3% of all intellectual disabilities for females in the world. So even though we're rare, we're actually quite popular. We hosted our first scientific conference back in 2015 with 8 families and only 40 known cases in the world.
Right now, we know of 1,307 cases across 62 countries. We know of 601 of those that are in the United States, 1,257 girls, 50 boys. Our Facebook group has 2,028 members, and we have 582 in our patient registry.
Here's where we are state-wise, which is pretty interesting to watch these numbers grow every year. And here's where we are in the world. So you can see, there's pretty much DDX3X everywhere.
We also are affiliated with several international organizations in Italy, Poland, the UK, Brazil. There's an overarching, Spain. There's an overarching organization in Europe. Gotcha. France, Japan, Australia.
Our approach to research is multidisciplinary. So we've been working very hard on animal models. So we have numerous mouse models you'll hear about today. We have iPSC lines. We've been working on clinical validation with our natural history study and our patient registry. And we have a big push on gene therapy for a therapeutic focus.
The research in progress right now, we have animal models. There are mouse models, which you'll hear about later today, that are being developed at Mount Sinai, Duke, UTSW, and the Jackson Lab, as well as iPSC lines that have been generated at UCSF.
And so here's a breakdown of the current animal models, and you'll learn all about these today. And then we are doing a biomarker exploration. And we're working on that here with Rush. And if your child is here, you can do that QR link, sign up while you're here, and get an EEG research for DDX3X with Rush. So the team can explain more about it as we get further along.
Research in progress, there's definitely been a therapeutic focus. Our primary focus is gene therapy research at UTSW, which you'll hear about from Dr. Gray today. There's promising data that has been generated recently, and there'll be exciting updates later today.
So here's a little bit of how we're doing fundraising wise. This year, we've raised $270,000 through event registrations like this event and the event in Arkansas. We pretty much break even for individual donations. So we've had some great gifts, and then family fundraisers, which we'll talk a little bit more about soon, corporate donations, and then online sales. At the end of 2024, we received an anonymous donation for $100,000 to fund gene therapy research, and that's been really helping us move that along, but we need more. So these are some of the family fundraisers that we've done. The Beast Feast, the Grace family's here, and they'll talk about that a little bit today. The Family Fun Weekend in Rogers, Arkansas, which is a great, just relaxed, easy way for families to get together and enjoy each other. Shores for Golf outing, which the Raider family's been doing every year. That's been really promising. The lemonade stand, a very simple concept that was very successful thanks to Sarah Golden and family, and Sarah's here today. And so enormous portion of our annual revenue comes from family fundraisers. So please don't hesitate to reach out if you're interested in hosting one and would like to support.
I also want to just take one moment. There's someone missing today, and it's Dr. Elliot Shear, who helped put us on this journey and connected Beth and I. And he was running a center of excellence in San Francisco. Unfortunately, for medical reasons, he had to retire. So he is still on email, and we're still in contact with him somewhat, but I just wanted to give him a shout out, because if it wasn't for him, Beth and I we definitely wouldn't have met each other. And so we want to just give our thanks to him.
Now, I would like to call up the Grace family. They have been incredible advocates and fundraisers for the foundation, and they have put together a video about DDX3X. And so I'll let them speak about that in Beast Feast.
Hello, all. My name is Spencer. This is Lindsay, and this is our oldest daughter, Avery. Our youngest, Emma, who has DDX3X, is playing in the other room. We wanted to talk about like how we got to this video. As mentioned previously, we've been fortunate enough to host an event. We call it the Beast Feast. If there's one thing that my family's good at, it's cooking, and we're really good at eating too. We come from a very, very rural area in Oklahoma. The town that we host our event in is less than 2,500 people. And so for us, over a 3-year period, to raise $140,000, it's significant, but obviously our people are very giving, and it's about, you know, life's about more than just us and money, and it's a way that we felt like we could do something to give back and try to promote research. So naturally, people will become involved and they want to help, and sometimes after the event's over, we get donations, and 100% of the donations up until this year have gone straight to the foundation. But we had some money trickle in at the end of the year, and I'm going to let Lindsay talk about what we did with that.
Okay, so our main purpose in telling you guys how big the town that we hosted in and how much money we raised is that we're doing it in a small town, and we raised a dramatic amount of money that we were more surprised than anyone. So I just wanted to encourage you guys that you can do it anywhere. You can raise the money anywhere with any kind of event that works for your community. So after the Beast Feast this year, I had an idea that we should save some of the money back and work on a video. And this was kind of late in coming, and we didn't have a whole lot of planning time because we wanted to get it in before the Family Day in Arkansas, so that the video team, because we did not do this ourselves, we had a crew that we hired to do it. They came to Arkansas, videoed families, interviewed families. We wanted more than just our little family in it. So we saved some money back, made this, and our hope is that you all will be able to share it wherever you want. We're not, that when any fundraiser, social media, whatever, we want this to multiply the donations. Any money we spent on this, we would love it to be way more worth it, 10 times, 100 times than we spent on it. So we'll play it for you guys, and we'll have it online. We'll share, I'm sure, on the website, or it'll be easy to access. No. Green one? Okay.
States that with DDX, we did not get a diagnosis. She was not results. We knew something was wrong. One was from birth. So for developmental pediatrician, neurologist, geneticist, specialist, it's up and down the East Coast. And when she started to speak, I said, she sounds like a deaf person. And so her to an ENT. And the ENT said, you're right, she can't hear in one ear, and the other ear is pretty clogged, but you and I both know something else is going on here, right? And he said, well, I've been going to this amazing clinic that made me remember why. It was a clinic for Amish and Mennonite children in rural Pennsylvania. I met with the most extraordinary doctor, Dr. Kevin Strauss, went to Harvard Med School, was a compassionate doctor. I would see to this. And so my husband and in Virginia, all gave blood, and they sorted through each of our 23 billion letters of DNA. Almost to the day later, they called me and they said, we know what she has. There's one of her.
Diagnosis for developmental delay, cerebral palsy, all kinds of other things. We just knew something was off. And so we went through all the other tests, and then in the last step, genome sequencing, just having.
You know, anybody that's you kind of have this idea of what your future would look like. You envision your kids growing up and what that would look like. You find out something's different. I think most parents agree there's a Do you agree when you go through kind of that lost feature, even though it wasn't there? Don't know what to replace it in your mind.
I will never forget that day. It was in the parking garage. And I was leaving the courthouse and I received a phone call from the genetics clinic. They said, we have a diagnosis finally. We've been searching for that diagnosis. We've gone to the medical clinic. We've done all sorts of testing. And I said, okay, well, what is it? And they said, it's a DDX3X. I said, hold on. And I grabbed a pen and paper and I wrote it down. I said, what does that mean? The one on the other side of the phone said, we don't know, but we can have an appointment for you in 3 months and we'll discuss it with the nurses. 3 months? What? And she said, if we find out some more information, we'll call you. But all we know is it's DDX3X. That's it.
Los Angeles, we wrote to say, so, meet with our neurologist, Dr. Elliot Shear. There was a genetic component involved. He told us that it was a rare disorder called DDX. Yes, they didn't know that much about it. And then coaching said, you need to start a family. Not only was I told that my daughter has a rare genetic disorder, then I was also studying homework. Fortunately, I was connected by amazingness and divine intervention to Beth Puccini, and so we around the world together.
I got my diagnosis in November of 2014 for Virginia. And so the Clinic for Special Children said there are 8 families in the US want to have a family day. And let's try to get as many families to come. So please come. I showed up. They explained it was a mutation of the DDX3X gene that causes intellectual disability. It was a really, really, really powerful day for this very team of us. There was one DDX3X child present that was just parents, DDX3X children. And at the end of that day, the genetic counselor at the clinic, who is now the operations director, said to me, okay, Beth, you need to launch a foundation. Make a registry, you can get families together and provide support, and you can find a cure. I said, I work in fashion. I definitely can't do that. She's like, no, you have the right person for it. There's another mom to do it with. And so introduced me to the other mom, and I said, work in science. He said, no, I work in Hollywood PR. It's like, this is ridiculous. How are we going to do this? But 2 hours later, on a phone call where we both cried, we committed to doing it. We launched the foundation in 2016, really, the website. We launched our website in February of 2016 with a patient registry with hopes of funding research and finding a better cure treatment for all of these amazing humans with DDX3X.
Children who had developmental disabilities for so long, there were so few conditions we understood, things like Down syndrome and those sort of things. With modern testing, literally thousands of new conditions being described that we never figured out without genetic testing. Challenges that people face DDX3X are not unique to that condition, right? That it's the bigger community of persons with developmental disabilities. And the challenges are, first off, just activities of daily living, right? Getting through the day, doing the things that I need done. All of us have to deal with that, and these patients need help. Second thing is integration into society. Again, over my 3 years of doing this, clearly society has got so much better as far as protecting people with disabilities, those sort of things, but there's still a long ways to go. And we still have to deal with the challenges of critical people, nosy people, judgmental people, all those sort of things. And so again, that's one of the things that support do. They raise that awareness. Most people are afraid of what they do.
I found out about the foundation through doing my own research when I found out proper diagnosis. And it had popped up that there was a foundation that created, and it was a blessing to know that there were people out there that have children have the same struggles. And just knowing that I wasn't alone.
This foundation needs help. It is the drugs and the research only come with money and support because we are a rare genetic. That means our numbers aren't big enough for large pharmaceutical companies to have interest or the government to have interest. So everything we do is upon donations.
Awkward to ask for donations. It's also hard for us to put into words where that's going. Can't say, hey, this is going for this next, medical research and getting to the next level and getting awareness out. So thank you. Every dollar matters. Scientists are doing everything they can, and we have only so much time in the day, so we would love to do more fundraising and contribute how we can, but I think so every little bit helps. We are thankful. My biggest advice would be if you have the funds and the ability to be able to support a nonprofit or us specifically at DDX3X, is to utilize those dollars to find some other rare genetic disorder or something that's close to your family, because progress will not happen without financial support, and rare disorders is the new norm.
Definitely softened my heart, honestly. It's having kids with that too. Having an DDX3X kid and profoundly changed life.
Hi,
You have to.
good morning. How was your day yesterday?
Heart changes.
Yeah.
With other people that you didn't notice with. But a lot of us are not going to be about
No.
fitness or being on a stage and attention. It's
Oh, well, that's good.
like clear love and say that's what you.
Oh, good.
Lily has taught me so much that love, because she is so incredibly loving. She just represents joy, and she is so happy all the time. And so me and my husband Alex get so much satisfaction, which is to be happy. And we didn't start the foundation necessarily to talk on girls, but we wanted to talk future generations.
That was pretty awesome. So we want to give a quick shout out to Speak Now Productions. They're a local production in Ponca City, Oklahoma. They put that together. It was started by two brothers, both in law enforcement. I myself am in law enforcement. So we have a strong bond, and I think they do a phenomenal job. They've really expanded. They do several of these types of videos for different nonprofits and other things. Thank you, guys.
Thank you. So now we're going to move on to the nitty-gritty scientific portion of the conference. So it is our great pleasure to introduce Tamar Caceres from Rush University and Stephen Floor, who is a DDX3X advisory member. They're going to give us a big talk about understanding the larger scientific picture about what we've been doing. So Dr. Caceres and Dr. Floor, please come forward.
Also, just want to thank the Grace family for that amazing video. You guys really knocked it out of the park.
Give us just a second while we set things up.
So I guess I just, okay.
Check. There we go. So I just want to say thank you all for coming. And for those who haven't met me, I'm Stephen Floor. I'm a scientist studying DDX3X. I'm also an advisor to the foundation. I'm excited to work with folks like Tamara and all the other scientists here. But what I really wanted to say is that, this meeting is for you all, right? It's, we talk to each other throughout the year. We talk to each other throughout the year. We have science meetings. This meeting is really for you all. So, the purpose of this session is to talk about the science and medicine that's happening around DDX3X. But if you have questions or things that you're not sure about, or you want to talk about something, please raise your hand and get involved. Tamara and I are professional speakers and could talk for a long time. But I think it's best if we, have a dialogue, and we're also happy to talk about stuff too. So.
Thank you. We're at different heights. So I'm Tamara Caceres. I'm a pediatric neurologist and neurodevelopmental disability specialist in Chicago at Rush University Medical Center. And so I take care of individuals clinically with DDX3X syndrome, and I'm also very involved in clinical research. So I was tasked with setting the stage for the day today to talk about what we will hear in the talks throughout the day and give context to a lot of the science that we spent talking about yesterday and talking about the different aspects that go into understanding a rare disease and linking the preclinical work like animal models to clinical work. So many of the priorities with regards to what we'll be talking about are priorities of foundation initiatives, and I'm going to go and we're going to hear from all of the researchers themselves. And so I can't do justice to talking about their work. So they will all individually talk about a lot of the amazing basic science and preclinical work. We will go through examples in other diseases of where these initiatives have led to progress in the field and what we can learn from them in terms of what's being done right now for DDX3X syndrome. I want to thank Dr. Barry Kravis, who I work with in Chicago and in clinical research in various neurodevelopmental disorders. And so she shared a number of these slides, and I'll also thank Dr. Shear, who is an amazing mentor in the field as well. So in neurodevelopmental disabilities, we can learn a lot from research in other syndromes as overall processes and critical steps are all very similar in terms of the processes that we use to understand rare disease. And so on this, I don't know if my arrow, yeah, I have an arrow. So on this side of things, we have all the critical work that's being done at the cellular and genetic level, looking at cells and at animal models, and we call all of this preclinical work. Like it's not yet in humans, but it's foundational in understanding disease mechanisms and overall helping us understand cellular mechanisms within a disorder. And then on the other side, we call this clinical work, like research in humans. And this process of getting from one side of the chasm to the other side of the chasm is what we call translation. So there's this big void and this big daunting task of how do we bring things and take the research from one side and understand how it's relevant in humans. And there's a lot of different elements of basic science research that we need to collect and understand in development of treatment for people. And each step in this process is really critical to understanding that. And so the foundation has really focused on trying to be pushing the field forward by targeting these different aspects that are all important. So you need biosamples and you need materials collected from individuals like blood and that's taken to the lab and looked at on the preclinical side of things. And so all of the families have really invested in pushing that work forward and fundraising and things like that. And so this is really relevant in all rare diseases in terms of how we understand that individual condition, but we can learn a lot from seeing how things have been done in other conditions. And in some cases, they have decades of work that we can look at and try to expedite, the process for DDX3X syndrome. So when we think about what needs to be done to be ready for disease-targeted treatments, animal models are really important to move drug development forward and treatment development forward. And we're going to hear about updates in animal models in DDX3X syndrome later today. Registries like the Foundation Registry are critical to understand like what is important to you, right? You may see a neurologist and not think that it's relevant that you have a symptom that isn't necessarily what you would think of as neurology. And so we don't necessarily know to ask unless you tell us or unless a group of parents all are experiencing the same symptom. And then that's communicated so that we can look at that if that is important to you. A clinical consortium, a group of centers that have experience both providing clinical care and or researching a condition, and it's important to, build these centers so that then families know where to go for care, but also then we have infrastructure to collect natural history and study outcome measures and run clinical trials. So we'll have another session later today where we talk about the natural history study and the progress that we've made with that. In outcome measures, looking for specific tools that we can evaluate a condition and understand the impact of future treatment. So we don't always inherently know what measure is going to be most appropriate to assess an individual based on their abilities and based on the spectrum of symptoms that we see in a specific condition. So we'll go through each of these and really everything that is happening to a certain extent within the within DDX3X research, and talk a little bit about examples from other neurodevelopmental conditions of how they impact the overall translational research process. So we can't develop treatment or targeted interventions without, oops. Is that something on my end? Stop sharing.
Family room.
All right, so while we work on technical difficulties, I'm just going to talk without slides, which is hard for you, for people who are visual learners, but I'm just going to keep talking. So with animal models, you know, as we are moving toward clinical trial readiness and disease targeted treatments, we use animal models to understand the mechanism of the disease in the brain and to try to understand things that could be measured using biomarkers and discover different treatment targets. So we can't develop treatment or treatment targeted interventions without really understanding the underlying disease at a cellular level. So we have researchers that are dedicated to developing cell-based assays and strategies to better understand the underlying condition, and animal models are one example of that. We can't take human brains and look at them, but we can look at neurons, like brain cells from animal models, and use that to develop an understanding of the disease and ways that we could potentially treat the condition. So we're going to be talking about animal model sessions in one of the follow-up visits. So an example of an animal model in another neurodevelopmental condition is in Fragile X syndrome. So in Fragile X syndrome in the 1990s, from looking at blood platelets specifically, they were able to identify that there was a specific cellular change in individuals with Fragile X syndrome. So they found decreased cyclic AMP, which is a specific specific molecule that it's a neural signaling molecule, and we found that in individuals who have Fragile X syndrome, there was decreased or absent FMRP. And then when you gave them too much of the gene that's affected in Fragile X syndrome, we found that you got too much cyclic AMP. And so at the time, there was no drug that you could target this mechanism in humans without a lot of side effects. And in the early 2000s, they were able to demonstrate that this was reproducible in different models. So in stem cells, in mice, in fruit flies, when you didn't have FMRP, the enzyme that is implicated in Fragile X syndrome, you had decreased cyclic AMP. And then when you had too much FMRP, you had increased cyclic AMP. And so we were able to, I say we, like collectively in science, not me personally. And so we were able to think about how could we use that pathway to develop a drug that potentially could target that. So about 8 years ago, or 8 or 9 years ago, a molecule was developed called zetalimostat. It's a phosphodiesterase inhibitor, and we found that it normalized cyclic AMP signaling. And so it was looked at in all of the different models, in the stem cell, in the neural stem cells, in the mice, and it normalized the shape of the neurons, and it normalized a bunch of behaviors in Fragile X syndrome mice. And so that foundational work really provided the data to take this molecule into human clinical trials. And so there was a phase 1 safety trial in humans, and it was found to be safe. And then they were able to do a phase 2 study and demonstrate improvement in cognition and in family ratings of challenging behaviors, in addition to improvement in an EEG biomarker in Fragile X syndrome. And that was the first treatment in Fragile X syndrome that was able to demonstrate that progress and progress to a phase 3 study. So we just finished the phase 3 studies in Fragile X syndrome of this small molecule. It's an oral medication, and they have now locked the data and are analyzing it, and they're in the open label part of the study. So the individuals are continuing to receive this medication while they are looking at the efficacy of the medication. So we're hoping for phase 3 results soon, and that wouldn't have happened without the preclinical research to inform the scientific rationale that was behind this drug. So we will switch over. It wasn't too bad, right? So we can see this is the molecule that we were talking about, and the how we sort of used early science to move it toward translation into humans. And so this is a lot more details about sort of the process of bringing it to a phase 3 clinical trial in humans. So on the, so it was, we first discovered signaling differences in the cyclic AMP system in the 1990s. There was no molecule that could target that system without a lot of side effects. So the molecule was designed in early 2000s, and then we didn't go into clinical trials with it until 2012, 2013. But the goal is to see how it happened and then do it faster. And we get better in different conditions at doing it faster once we figure out what we need, and how to learn the challenges of what things go wrong. So there are limitations to using animal models, right? Animals are not humans, and the model of the disease in the animal doesn't always translate to what the human looks like. Some targets in the brain might be conserved between mice and man, but for others there's divergence and there's benefits in mice that may not be seen in humans. And so of course I have an example of that as well, and we can learn from that. So they also spent decades of time researching mGlu5 blockers in Fragile X syndrome, and extensive preclinical data showed that we could reverse symptoms in mice using these specific type of molecules. And a lot of research was focused on targeting this molecular pathway that is dysregulated in Fragile X syndrome. And they got all the way to phase 2, phase 3 placebo controlled studies. And in 2 separate 12-week targeted studies in humans, they were not able to demonstrate behavioral benefits. And so we're really able to, understand that we can't put all of the eggs in one basket. We have to think about how models in animals have symptoms or behaviors that may or may not translate to humans, and explore different pathways that are trying to target symptomatic treatment or cure using different strategies. So shifting slightly to the clinical side of things, when we are exploring how these different targeted treatments may impact the course of a condition, first we have to understand the condition. So we have to understand the range of symptoms, we have to understand the range of severity, we have to understand how things may change over time, and we have to have tools to measure that. So we don't always know exactly how to assess language or how to assess motor challenges or how to assess certain types of behavior in certain populations. And so we have to test the tools to see how they work. What do they look like in general in that population? So that if we tried an intervention, how would that change what we know so far? And so through information that we collect in the registry, where we find out what's important to families, what symptoms are you seeing, through standardized assessments, trying out these different tools, we can make progress toward understanding what measures we might be able to use in things like clinical trials. So right now, UPenn and UPenn CHOP and Rush are recognized as centers of excellence and for DDX3X syndrome. And there are lots of examples of how clinical consortia or centers of excellence across different diseases have been helpful in the translational process. So different sites might be focused on research or clinical care or a mix of research and clinical care. And there's value in being able to have designated sites so that individuals both have an opportunity to see providers who have experience taking care of children with that condition, but also then we have large groups of people going to consistent sites, and we're able to harness that in terms of exploration of research and different types of clinical trials. So these are just examples of other clinical consortia that exist for other rare neurodevelopmental disorders, and they all have different sort of structures of what's involved in their centers of excellence. So scientific advisory committees, medical advisory committees, clinical care, development of practice-based guidelines, natural history studies across all of these different disorders. I lead the natural history study at Rush in DDX3X syndrome, and we'll talk more about that this afternoon. But the goal of natural history studies, as I discussed, is to systematically collect information about a specific population to establish the range and severity of the disease and look at change over time. So we have a lot of really great cohorts in DDX3X syndrome described in the literature to help us understand the types of symptoms. But each of those cohorts uses a different measure or battery of tools to describe the population, and they're all single assessments in time. So the individuals are not necessarily seen multiple times. And so our goal is to follow individuals over time to see how to better understand developmental trajectory. We have lots of families that, say that their kids are still learning amazing things at 8, 9, 15, 20, and we want to understand what that is. And are there certain subgroups within the population that would potentially be the best candidates for certain types of therapies and the timing that we would need to evaluate efficacy of therapies, right? Do we need to evaluate somebody after 8 weeks? Do we need to evaluate them after 3 months? Or do we need to evaluate them after a year based on the rate of change that we expect? So there are various examples of how natural history can contribute to better understanding and clinical care, right? What are the recommendations for the individual based on their predisposition and underlying genetic syndrome? So we have a lot of this for DDX3X syndrome already based on expert opinion and collaboration and what we know in terms of is there medical surveillance that's recommended for care? And so using natural history study to continue to expand on that information. Feel free to jump in with questions. So we work on developing and understanding phenotype-genotype interactions, right? We know in DDX3X literature that there may be some genetic changes that have a different clinical picture than other genetic changes. And so the more information that we can collect on that helps us best understand things and bring that to the in-human clinical research side of things. You can better understand other organ systems, right? So I'm a pediatric neurologist. We try to consider the whole person in our care, but we don't necessarily know if there are symptoms targeting another organ system unless we are seeing it over time and following those individuals and hearing from families what symptoms they're experiencing. Yeah, absolutely. So So there are different setups for different foundations. So typically for individuals who are coming to one of the centers of excellence for clinical care or consultation, that is billed through insurance. And so there are different processes for different people for out-of-state insurance and things like that. We're available for questions or things to run by us, and the foundation can certainly connect you with people who are actively involved in seeing patients.
But there are also a couple, I mentioned earlier, I think there's a couple papers that are directed at clinicians, right, that do have recommendations about symptom management and care for different classes of symptoms. And that's another place that clinicians that are just learning about DDX3X can turn to, is to read some of those papers, including some that you've been involved in those too.
Absolutely. And the goal is to put more out, right? So that we can use this information to better inform individuals who may not have specific experience, but have the ability to help understand the guidelines. So another aspect, in addition to registry and natural history and animal models, is looking for potential biomarkers. When we say biomarkers, we're meaning things that we can objectively measure, either in the blood or using EEG, neurophysiology, to look for patterns in a condition that we could potentially track and see if it changes with some type of intervention. So we are just hoping to start exploring this in DDX3X syndrome. In Angelman syndrome, we found that there is a pattern in EEG called delta power that's abnormal in Angelman syndrome, and it predicts cognition. So individuals who are more severe in terms of their cognitive function have higher delta power, and in some of the targeted treatment trials, they're able to demonstrate that there is improvement in that. And so in addition to the clinical measures and clinical tours that we're using to assess their abilities, we're also able to look at how it impacts a biomarker. So we are starting to try to explore this in DDX3X syndrome. If anybody is here with their child with DDX3X syndrome, at the conference, we have all of our EEG equipment, and Jordan Norris is our postdoc fellow who's working on this project. And so this is a parallel project to the natural history study, and we'll talk about it more later because it's not the focus of this talk. But if anybody's interested, we can talk to you more about that process. It's not to look for seizures or to inform clinical care. This is specifically looking for patterns on the EEG to inform research and understanding of the neurophysiology of individuals with DDX3X syndrome. Yeah. So the question was, if your child isn't here, how might you be able to participate in this study? So you can always come to Rush in Chicago. Hopefully we'll be back every year to do this. And we can also explore options for other sort of periods of time. It's, a non-invasive, very scalable tool, right? We were able to bring all the stuff on the airplane with us. And so this is not a, the opportunity is available. It's not just today, but we wanted to take the opportunity while we have a bunch of individuals here, if anybody is interested. Not right now. Yeah. Yeah. So chat with Jordan and Melitza, and we can work on coordination. Unfortunately, it cannot. Yeah. So the question was, can the data be extrapolated from existing EEG results? When we are doing research EEGs, we are looking at the EEGs with greater detail than what is collected on the clinical EEGs, and it's not from the report. Jordan actually looks at all of the brainwave data and is looking at patterns that are different than what we're looking at when we are exploring clinical EEGs. But I'm going to circle back to this, and we are available, and I'm glad that there's so many questions and excitement. So in terms of outcome measure development, we talked a little bit about this. For anybody who has participated in the natural history study, we're having people do lots of different parent questionnaires, and we are having individuals doing lots of detailed performance-based assessments where we're looking at their motor skills and their language skills. And again, we're trying to explore what is important to look at and what tools can we use to assess people across a wide variety of symptoms and severity. In addition, typically in clinical trials in rare disease, tools are developed specific for that condition based on what symptoms that condition presents with. So identifying different domains that are affected and developing clinical global severity scales unique to that condition are often part of the process of developing clinical trial readiness and utilizing those scales in clinical trials to support evaluation of efficacy of an intervention. We know that, in genetic neurodevelopmental disorders, we're moving toward it being a field of more than just supportive treatment. So we have exciting preclinical and clinical work in many different types of therapies in terms of antisense oligonucleotides, gene therapy, what we call small molecule interventions. And so we are, excited to be collaborating and working toward being prepared with strategies that that can help prove that these treatments work or don't work, so that we know. So we'll open it to questions if we have time for questions, and if Steve wants to add anything.
Yeah, I just want to emphasize what Tamar was just saying. So there was a recent trial for a different gene therapy in the eye that was run by Johnson & Johnson, or Janssen Pharmaceuticals, where they picked an endpoint for the clinical trial that was a little unusual. And then they had these secondary endpoints. The way that you register these clinical trials is that you decide ahead of time, right? This is what we're going to measure. This is how we're going to intervene. And then at the end, you do the measurement and it either succeeds or fails. And you might have a bunch of other backup stuff that you're looking at, but there's really this one primary endpoint that is really the determinant of whether it succeeded or failed. And this is for ethical reasons, right? You don't want to go back and say, oh, actually everything was great, right? And so they picked this unusual outcome measure and they failed at it. And they picked a more common outcome measure as a secondary outcome, and that succeeded. But now they can't go back to the FDA and say, oh, actually, guys, this other one is what we really should have looked at. It doesn't work like that, right? So the registry and the natural history study is extremely important because not only do we understand about progression and how people develop, but it equips clinicians and scientists to actually run clinical trials with the endpoint that's the most likely to succeed.
Parallel. Studies approaching the topic from different angles.
Talking to the mic so they can.
Sorry. It sounds like there are a lot of studies approaching the topic from different angles. And I just wonder, is there a scale or volume of patients that you need in order to make the results of a study viable? Like, do you want a certain number of us to be enrolled in a natural history study in order to make that data useful? Is there a certain number of us that have to be on the registry? Just how much should we be mobilizing to reach that threshold?
So, I mean, we would like everybody enrolled in the natural history study and everybody signed up for the registry, right? Because that gives us the best chance of having the tools we need. Certainly there are, goals to make that more feasible, right? Like it is hard for families to travel in person for multiple days and pay for that out of pocket. And we recognize that. And hopefully as the field moves forward, we will have more financial support for that and be able to try to make things as accessible as possible and help us scale the project, right? So that we have more structure on the database side of things to be organizing and interpreting and evaluating that information. Do you have other comments on that? So yes, like the registry, you can fill it out online. Like hopefully everybody is enrolled in the registry, right? And we're working on trying to increase the number of sites for natural history and potentially do a hybrid process so people who can't be evaluated in person may still be able to contribute and things like that. Go ahead. Or maybe wait for the mic and then go ahead. I'll bring it to you.
We are newly diagnosed, so I did complete the registry, and I was wondering the categories in the registry, like are those finite, or do you add categories over time as you learn more, meeting more children with this diagnosis?
So the registry is the foundation's registry. So I'm not directly involved in what's in the registry, but I imagine there's a registry team and you should absolutely give feedback.
Yeah, absolutely. So it does kind of evolve over time. It's ongoing. We encourage people to go back and, update data. We add questions. So if you have thoughts, please do let me know.
Just because she's 40, so we're looking at other issues, as she
Absolutely.
ages. So yeah, I'll go back for sure.
Perfect. Thank you. Sure. Yeah. So one thing that we're going to update in the registry, we got some feedback yesterday during our research roundtable meeting. One of our researchers had asked for the de-identified data, which is part of the purpose of the registry to provide researchers with the information that we have available. And whenever this researcher started looking at it, they found that there were a lot of inconsistencies in how people were reporting their variant. So that's something else that you may be hearing from me. We'll be going back and updating that to make that information usable to researchers. And we will be adding some more clarity about exactly where to look on your genetic report to find what we need. So that's also just another example of how things kind of evolve over time as we receive feedback from our clinicians and researchers.
And we know that there are probably lots of adults with DDX3X syndrome, and we know the least about that population because it was only discovered, approximately 10, 15 years ago. And so we will constantly be learning more about the adult symptoms that may affect that population and need to add in and collect as we go. So that's super valuable information, and updating the information in the registry, right? The symptoms that you are having at the time that you complete the registry the first time change. And so they want to collect that information over time as well.
And one thing I'll say about the older patients is that we are very close. We're just in the waiting IRB approval for a Center for Excellence in conjunction with both CHOP and Penn. And so we're focused on some older patients going there, young adults and older adults as well. So that's going to be a focus there because at Penn, they're used to older patients.
And at Rush as well, we are happy to see adults with developmental disability.
Hi, slightly different question, but I do work in data and analytics. So I'm wondering if recent advances in computing processing power, AI, is that helpful to this research, what you're doing at all?
Absolutely. I'm a pediatric neurologist. I have no experience on that end of things. And so harnessing all of your skills can be super valuable in helping advance different aspects of this research.
Yeah, I just would add that, one of the, and we were chatting about this earlier as well, I think one of the, and yesterday, one of the challenges is that you want to analyze all the data and you all have a ton of data, but we also have to do it in a way that's ethical and up to clinical standards, right? So, you can't just email all your data to me and I put it in my Dropbox and write some code and see what happens, right? Like we have to make sure that we're doing things, ethically and protecting patient information and all that kind of stuff. So we try to balance those two, right? That, those can be barriers because we have to make sure we have the right processes in place to protect the data and are up to code with dealing with clinical data, but we also want to analyze as much of it as we possibly can. So there was a lot of conversation about that yesterday, and I think we'll carry it forward. But that is kind of one of the challenges when dealing with clinical data.
What I'm referring to about yesterday is we host every day before the conference when the doctors, scientists, and researchers present to all of us. We host a scientific roundtable where we put everyone in a room who's studying DDX3X, and we go through what advances we've learned in the past year and what's on the horizon. So.
Okay, we have time for probably one more question, and then we're going to take a little break.
Years, our daughter had autism and now she doesn't, or maybe she does, I don't know, but she has DDX3X. So what's being done to get more people genetically tested in terms of like reaching out to outreach to providers? It was not pushed upon us. It was just our own.
Yeah, so certainly there has been a shift as we have come to better understand the role of genetics in intellectual disability and autism spectrum disorder to integrate into training of medical providers in pediatric neurology and developmental medicine, in psychiatry, in pediatrics. And so now it is standard of care to do targeted genetic testing, typically exome or genome, first line now for individuals with global developmental delay and or autism spectrum disorder. But that has changed over time. So previously it was a type of genetic testing that wouldn't have picked up DDX3X syndrome with chromosomal microarray. And so, but this is a process and it takes time to train clinicians and then do updated trainings. And so that's why we have a lot of undiagnosed individuals. There's also a barrier to access to care, right? A wait list to get in to see somebody who specializes in that area, maybe 18 to 24 months, or there may be nobody in your area, or it may be outside the scope. So that is still absolutely something. Cost, exactly. So that is absolutely something that we are working on, right? When we are eventually have targeted treatments for some of these conditions, pushing to have it on the newborn screen. That is something that is state dependent. And so there are all different factors that can contribute to the diagnostic odyssey for people with developmental disabilities.
Quick question. So we were connected to UCSF for the data research, and we were wondering in terms of the status of that, if that's being absorbed by another study, and how do we get involved with another entity if we need to?
So, and we'll talk about this a little more later, but for anybody who enrolled in the natural history study at UCSF, it's all part of the same study. So we have that data. There is a little bit of a process in terms of coordination and timing, but you absolutely will get updates and you'll be connected with one of the current natural history study sites to coordinate and hopefully arrange your follow-up, although we understand that there is a burden with that. We saw that there's a large number of individuals in California, and so the site won't necessarily be as close to you, but your data is safe, and we would love to have your ongoing participation if that's feasible for your family.
Tamar, could you explain how it took us over a year to get everybody on the same page in terms of having the same set of tests that we were going to do, and so it's standardized across the board?
Yeah.
Tamar was the big boss of that. So
Yeah, so this afternoon there is a specific session focused on the natural history study and updates from the natural history study with a QR code to contact Melitza and all of those details. There have been many different amazing research projects focused on DDX3X over time. So there have been people that have participated in projects historically at UCSF. UCSF was involved in the natural history study. And so we're happy to talk with you about the details of that and what that means and what's going on now that would be opportunities for you to participate in. And for the natural history study specifically for the people who have been seen at UCSF, how you can continue your participation, which we hope you will.
Okay, and with that, I think we're going to break until 10:45 when we'll get back and talk about some of the science. Thank you all.
And thank you to Dr. Caceres.
There's no HDMI.
All right.
Give you some chairs there. Thank you. Yeah, but these blue lights gonna heal us, but. Yeah. That's pretty. Yeah, we're just because of the blue light. Blue light, exactly.
Everyone could please have a seat. We're going to start up again. If everyone could please have a seat, we're going to start up again.
Okay, we're gonna get started now. We're gonna move into the science session, first of two. In this session, we have 4 presenters that are gonna talk about different parts of the research that's happening on the DDX3X gene, what DDX3X is, how it works in cells, how it connects to animals, and what it is that we can learn about different things that are happening to especially animal models that tell us about what's happening to people that are affected by DDX3X. So we're going to have 4 presenters in this first session. We have Debbie Silver, Sylvia de Rubiz, Andrea Boitnot, and Jen San Miguel. We're going to go in that order, trying to build up from the basics into more animal behaviors and then new animal models. And then after that, we're going to have a session with Stephen Gray that's going to talk about gene therapy. So how can we build on our understanding of what DDX3 does in animals to try to intervene and correct some of those errors? So we're going to start with 4 presentations, and I'd like to hold questions until the end so that we can get through those, and then we can ask questions of everybody at the same time. So we have 3 folks that are here, and Jen's going to join us by Zoom. I see her there. Hi. So Debbie, you're up.
All right, so really a pleasure to be here with you all. I want to start with this introductory slide. This is the team of researchers on the left that have participated in our work for many years to better understand how different types of mutations in DDX3X impair the development of the brain. And then I want to particularly highlight the folks here who are the most recent lab members whose work I'll highlight. And so our lab is largely interested in understanding how different types of mutations in DDX3X can impair prenatal brain development. And we think that this is critically important because this can help us to learn better about the earliest causes of the syndrome, and in particular, different types of parts of the brain that are affected, as well as or other organ systems, as well as types of molecules that are affected. Uh-oh. What just happened? Okay. And so the other rationale for studying the development of this disorder, and we heard a little bit about this earlier, is we can start to identify commonalities with other models that are affecting neurodevelopment. And so all of this, of course, is really important for us to gain a basic understanding of DDX3X syndrome, but also to ultimately have the goal of designing effective and very specific therapeutic strategies. And so I want to start with this really simplified schema and remind many of you in the room that that your body is composed of billions and billions of diverse cells across different parts of your body. Within those cells, we each have chromosomes, and within those chromosomes are actually made up of DNA. And when we talk about mutations in DDX3X, that's where those mutations are happening. They're actually happening within the DNA, and it's throughout every cell of the body. Now, that DNA is critically important for encoding specific functions that make our cells work, that make the brain a brain cell, or a heart cell a heart cell. And so it does that by making something called RNA, and then going on to make protein. So the reason I am also showing you this slide is because DDX3X, as many of you may be aware of, itself is a protein that controls cellular functions by controlling the levels of other proteins. And so we kind of think about it as being a really master regulator in terms of those functions. And so I want to now highlight a few of the different types of mutations that can cause DDX3X syndrome. And many of this may be familiar to many of you. So about half of the mutations in DDX3X are thought to be nonsense. That is, they are predicted to make overall less of the DDX3X protein in the body. And so lower levels overall than what somebody who is not affected by the syndrome would have. And then the other class of mutations are referred to as missense. And these are essentially tiny pinpoint changes within the DNA that cause very select changes in the protein that we predict it makes it functioning less well. And that can be mutations that affect the protein's ability in a very mild way, or proteins that have a much more severe detrimental impact. And I think this is one of the really key questions that many of us scientists in the room are trying to understand. How do these diverse types of proteins ultimately influence the presentation of the syndrome? And so we in the lab have been tackling this question by using a multi-pronged approach. And what I mean by that is we use mouse models that we'll be hearing about today, but we also couple our investigation by investigating how different types of changes in DDX3X influence cellular function, and then using really cutting edge methods out there, technologies that allow us to discover what are the key proteins that DDX3X controls. And we can do this sorts of approach at very different levels. As I emphasize, we're in the lab really largely focused on prenatal development. And you'll hear from in the next coming talks about important functions of DDX3X postnatally. And then, of course, as I indicated, we want to be able to couple these approaches to not just a single mutation. There are very diverse and distinct subtypes of mutations. And so our goal is to be able to understand how diverse variants in DDX3X have a functional impact. And so one way that we've tackled this is by evaluating mouse models in which we can use engineering methods to generate mice in which there are overall lower levels of DDX3X. And that's going to be more like a loss of function or a nonsense mutation. Recall that was the type of mutation that was at the very bottom of that cartoon I showed you earlier. And then we can also model missense mutations. And so far, we've been evaluating a mouse model in which we engineered in one of a variant that is associated with more clinically severe outcomes. And so we can generate these mouse models, and then we can start to look at the different organs of those mouse models. We've so far highly focused on looking at the brain. The brains of these mice actually at a very high level anatomically look relatively normal, which is great news. But what we then try to do is to go inside those brains and to try to understand what are the cellular and the molecular changes that are maybe occurring. And so we can do that by a number of methods in the lab. And one cell type that we have specifically focused on are neurons. These are thought to be the building blocks of the brain that are critically important for controlling brain functions. The way they do that is by establishing connections with other neurons in the brain and helping to connect different parts of the brain to each other to make our complex brain functions proceed properly. And so a key aspect to understanding this is that we know that these neurons, they are actually made during prenatal stages of development. And so using approaches in my lab, what we've discovered is that DDX3X mutations have an impact on the number and even potentially the sort of type of neuron that's actually made. And this can be help us to better understand how impaired brain function is leading to DDX3X syndrome. And so I want to show you a movie here in which seeing is believing. So we can take cells of embryonic brains and we can put them into a tissue culture dish and essentially make movies of those and watch those progenitors divide and see the extent to which they make new neurons. So this is a stem cell, it's dividing. It's making 2 red cells, and those red cells are what we would call newborn neurons. And so we make lots of these different types of movies. We can make them using cells that have reduced levels of DDX3X or cells that have some of those different types of missense variants I described earlier. And so we make complex graphs like this. Reading this is not important, but what I do want to emphasize is that from doing these sorts of analyses, what we've begun to discover is that distinct types of mutations in DDX3X will subtly reduce neuron number, but you can see and hopefully appreciate that there are different classes of how these different variants behave. And I think that's really important to emphasize because, and you all know this from the clinical presentation, not every case of DDX3X syndrome presents identically, of course. And so we think it's really important to be able to investigate individual mutations. And one of our major goals is to be able to ideally begin to classify these mutations so that we can make predictions about their pathogenic impact. And then finally, I want to mention that we're using these sorts of models, these mouse models, as well as cell-based models, to discover key molecular targets of DDX3X in the developing brain. So DDX3X, shown here with this little blue blob, is essentially an RNA binding protein. So it binds to this type of molecule in all of our cells called RNA. And in doing so, as I mentioned earlier, it is critical for helping to make protein. And that protein goes on to serve all sorts of cell-specific functions. And so what we're doing is using these different engineered models in the lab to discover the key proteins that are disrupted when mutations in DDX3X are present. And so the idea then, of course, is that we can use these targets to better understand how DDX3X impairs brain development. We can also compare them across different developmental stages, different cell types that many of my colleagues are generating similar data sets for. And then we can ideally, of course, begin to identify converging and potential therapeutic targets. And so I'm going to stop there and hand it over to Dr. DeRubes.
I'm gonna do this. So hi, everyone. It is a pleasure to be here once again. We had a great session yesterday with all the scientists and researchers, but we're really eager to transfer some of that knowledge to you and have a discussion with all of you. So my name is Silvia De Rubeis. I'm based in New York City at the Icahn School of Medicine at Mount Sinai, and my lab entirely focuses on DDX3X syndrome. We started in 2017, and I always like to show this slide where I sort of place all the lab members across the mouse brain to show you the different projects. And everyone you see on this slide, but Adele, are here in this room. So you are welcome if there is any project that you think is you're particularly curious about, you're welcome to, touch them and discuss with them. So we have an instructor in the lab, Marta. We have a postdoc, Geronimo. I'm going to be talking about part of his work. And then we have 3 PhD students, Nunzio, Corinne, and Lauren. So we have heard from Debbie what is the use of mouse models, right? So mouse models really allow us to understand the biological underpinnings of a disorder, and being able to go from behaviors to molecules and back. And in the case of a neurodevelopmental disorder, we want to do that through a developmental lens, right? We want to keep the developmental perspective in mind. We've heard a lot about prenatal development and critical molecules and critical cells, but today I would like to focus on behaviors and our approach to identifying the mechanisms that are driving those aberrant behaviors in our mouse model. So I'll be telling you, I'm going to be giving you just 2 sort of vignettes of our work. Again, we're going to be here through the day, so happy to discuss with any of you about the extended body of work that support what I'll be showing you. So the first story is from Dr. Geronimo Lukin, who's sitting right there, who joined us all the way from Argentina to be doing these studies on DDX3X syndrome. And then the second story will be from Lauren, a PhD student, who joined us all the way from Connecticut to be doing these studies. Okay, so let's start with Geronimo. So what Geronimo is very interested in is what is driving anxiety-related behaviors in the patients with DDX3X syndrome. And this starts with an observation we made early on. Actually, Andrea, was sitting right there, was in my lab at the very beginning. So this is an observation she made in this paper that was published a few years ago together with Marta. And so when we approach understanding anxiety-related behaviors, I want to make very clear that anxiety is a human construct, right? We cannot ask mouse, how do you feel about, exploring this new environment? So we take ethological constructs, behaviors that animals would typically have in nature, and extrapolate behaviors from these paradigms. So this is a very naturalistic task. The animals are simply going to explore a new environment. They're going to be placed in an open arena. And what a mouse would typically do would be to avoid the center of the arena where they might be more exposed to predators, right? So what we saw, and this is published work, as I said, that when we look at the traces that an individual mouse has, so imagine you're having a bird view, right, from the top of the arena, and these are all the movements the mouse is making. So this is what a control female mouse does, right? Explore the arena, go to the center, not so much, but exploring the arena. And this is what a DDX3X mutant female is going to do. She's going to be, this is a mouse, right? So she's going to be sticking to the walls of the arena. So this behavior is called increased stigmotaxis, and it is used by researchers as a proxy for anxiety-related behaviors. So when we saw this, what we wanted to do is, what are the factors that are driving these sort of excessive response to a novel environment? So the approach that we take is that we expose these animals to this novel arena just for 10 minutes, and then we take their brains and we clear their brains. So we make their brains optically transparent. You can see a brain before we clear it, and this is after, right? You can look through. So what this allows us to do is to explore cells and patterns of activity throughout an intact brain. We don't need to slice the brain up. We can look at things in 3D. And so this is a view from a top of a control brain and a DDX3X mutant brain. You're seeing the full 3D. And every white dot you're seeing here is a cell in the brain that got activated, responded during the exposure to the arena. We can then register all of this information on the neuroanatomy of the mouse brain and identify specific regions in the mouse brain that are either activated too much or too low. What we generally see in the DDX3X mutant mice with this paradigm is that there is an overactivation of regions in the outer shell of the brain that is called the cortex. So then what Geronimo can do is take other cohorts of animals and selectively express artificial receptors that are going to drive the activation of a specific brain cells on or off. It can literally put this virus in the mouse brain and switch on and off the activity of some parts of the brain. So in this case, what he was able to do with this work is identify a brain region that is excessively activated by the task. And this is just showing you one of the many paradigm measures that we look at. This is latency to enter the center of the arena, how long it takes for a mouse to decide, okay, I'm gonna go to this open space that is very risky to me, right? So I hope you'll appreciate that it takes them longer. These are the DDX3X mutant females, right, in red. It takes them longer to enter the arena. They're very hesitant, right? Again, reminiscent of these anxiety-related behaviors. Now, when Geronimo switches off that specific region of the brain, what he sees is that these enhanced anxiety-related behavior is now ameliorated. So what he's going to do now is figuring out what are the specific molecular drivers that are driving, as they are drivers, that are leading to these ameliorated behaviors. So this is one example. I'm going to give you another one. This is work by Lauren, who is very interested in motor development, which we all know is impacted in DDX3X syndrome. And we are also interested in motor behaviors from a more developmental perspective, because very early on during infancy and early life, ability to navigate the space through motor skills is very important for the interaction with the environment and other people. Plus, motor skills build one upon another. So very early delays can have sort of cascading effects on development. So what Andrea and Marta had characterized in our mouse model is that we really have a altered trajectory of motor development in these mice, starting neonatally when they show hypotonia and then motor delays. In juvenile age, they show abnormal gaits. When they're adults, they show impaired motor skills in a variety of tasks. And then with aging, they show a motor decline. So as part of Lauren's project, she wanted to know whether we could identify a strategy to change this trajectory, right? In other words, can we intervene early on during the mouse life to make them, to sort of prevent or ameliorate this trajectory? So what she came up with is early life exercise intervention. So what she's doing here, she's taking control that you're going to see in blue and DDX3X mutant females that you're going to see in red, and either leave them in their cage during the juvenile age or have them basically go to the gym, right? For 5 weeks, once a week, she places the mice on a rod and she helps them learn how to do the task, right? They are on the rod and they have to walk on the rod. So, and then 5 weeks after, she checks for their ability to perform this task when this task becomes a bit more complicated because the rod accelerates. So what you're seeing here in this plot is the latency to fall off the rod. The better you are at the test, the more you can stay on the rod. And these are our control groups, right? Mice who did not have the intervention. As you can see, our control females typically do better than our DDX mutant females, and that's in keeping with their motor impairments. But in the group of animals that underwent these early life intervention, then the motor performance was much better, right? You see them, they can stay on the rod much more. Lauren also wanted to understand whether these benefits would persist with time, and so retested the same animals at 4 months of age. And what she saw again was that there were long-term benefits of the intervention, because now these mice, months after the training, were still better at performing the task. Why is this important? Now Lauren can take these brains and figure out again what are the factors that have changed with this early life intervention, and maybe these very same molecular factors can be targeted pharmacologically. Okay, I'm just gonna close there. This is the team. As I said, we're based in Upper East Side in New York. Everyone is welcome to join us in case you wanna see the mice. We have had a few months joining us to check out the mice, and I wanna thank the DDX3X Foundation for having us, all of you for bearing with me through this presentation, and I'm happy to take any question. Thank you. Oh, we said we're gonna do questions at the end, I think.
Yeah, let's
Okay,
move on
thank
to
you.
Andrea, and we'll do them all together at the end. Thank you.
Okay, I'm gonna start with a video of one of the mice that we have in the lab. Here you'll see the mouse walking around in its cage. This is where it spends its whole life pretty much. In one of the cages, you'll see its water and food, and he made a little, actually this is a female, so she made a little bed over there in the corner. This mouse you'll see has like that kind of like side backpack. It's a sensor that we're using to quantify some behavior, and that's behavior is sleep, and I'll talk about that in this presentation. But I guess if you just keep zooming through this video, this is a video that is taken over a whole weekend, and right now it's sleeping. So we can differentiate when the mouse is awake and when it's asleep, and look at different behaviors between our DDX3X mice and our control mice. So let me go to... Okay, so I'm going to talk about sleep disturbances in our mouse model of DDX3X syndrome. I'm Andrea. I've met a lot of you before. I come to these every year. It's one of my favorite times of the year to come see all the families. And okay, I graduated from Bates in Maine, and was my thesis project in undergrad was on learning and memory and intellectual disability models. I really got interested in neurodevelopment and the rare disease space, and I was looking for very specific jobs out of undergrad that had to do with pediatric disease, neurological disease, intellectual disability, and I found the perfect job description, which was in Sylvia's lab. It's the only job I applied for. And it is, I had no idea it was in New York, moved to New York, worked for her for 2 years, developing some of the preliminary research on the mouse model, behaviors in the mouse model of DDX3X syndrome, wanted to transfer into the therapeutic space, so applied for my PhD moved to UT Southwestern to work with Steve, who's here, and will give a presentation on the gene therapy work, and took on DDX3X as the project to try to work on some of the initial development for a gene therapy. So that's my background. I've been in the DDX3X space for, I think the first DDX3X meeting it was 2019 that I went to, and I've been there every year since. So yeah, so the sleep disturbances. So in the video, you saw that one of the mice that are in this home cage. So we can measure sleep by recording brainwave activity, EEG, and also muscle tone, EMG. And we do this with a wireless sensor. So the sensor is what you saw in the mouse. It's like kind of like a little backpack that's implanted right under the skin. And then there's wires that go to the brain and to the muscle, to the neck muscle. And from that, we can record these EEG waves and EMG signals to understand when the mouse is awake and when the mouse is asleep. And this requires a pretty intense surgery. And we'll perform this surgery about a week and a half before we record the mice. And we record the mice over a weekend. So we have continuous data on when the mouse is naturally sleeping and when the mouse is naturally awake. And that can be very helpful for understanding normal behaviors in these mice. This is the data that we get off of this sensor. And from this, we can differentiate two phases of sleep and wake. We can differentiate slow wave sleep, which is like our non-REM sleep, and paradoxical sleep, which is equivalent to our REM sleep. And you'll see the trace. This is just one part of the trace of the whole weekend scoring that we do. And you can see when I'll maybe direct your attention to this black bar here. When it's mostly black, like that's when the mouse is awake. And then when you see these like white bars, that's when the mouse, maybe I can point to it. So this is when the mouse is mostly awake and moving. You can see its activity. And then these white bars are when the mouse is asleep. And then there's intermittent times when the mouse transitions from the slow wave sleep or the non-REM sleep to paradoxical sleep, which is the REM sleep. And you can also see that within this like sleep bout with like those little dots of white that indicates the REM sleep. So we have a ton of data, and then we can bucket this into when they're sleeping in these different stages and quantify and ask many questions. So we have the trace, the yellow would be the wake, purple is our slow wave sleep, and red is the paradoxical sleep. And then we can ask like how much time do the mice spend in each stage, and the number of transitions to and from each stage, the number of bouts for each stage, the average bout time for each stage, and the shortest and longest bout time of each stage. And just to maybe a little bit more background, from coming to these meetings, I hear a lot from parents about sleep issues that they in their kids. And one of the things that I try to do in the lab is find behaviors that we also see in the mice. So we have behaviors in both the human population and the mouse population that we could use to study maybe different mechanisms or use for when we have therapeutics that we want to try to see if they work. So we could intervene and see if it has maybe an effect on sleep in a positive way, in a beneficial way. So the results from this study showed that we had a sleep phenotype in our DDX3X mice. And I'll just maybe direct your attention to the top bars here. So in the control mice, again, the wake is in yellow, and the slow wave sleep is in purple, and the paradoxical sleep is in red. And in our control mice, we have maybe these longer times spent in the wake phase, a little bit less time spent in the sleep phases. But we also have like more maintained sleep. So in our DDX3X mice, the mice will transition from slow wave sleep to paradoxical sleep and back to slow wave sleep at a higher frequency and shorter times in paradoxical sleep than the control mice. So it's really indicative of the fact that the sleep is not as the sleep quality is not as good, and the paradoxical sleep specifically is not as well maintained. And we saw some other differences as well, but hopefully this will help. I think Tamara will talk a little bit about it in her later talk on the natural history study, but they've introduced like a questionnaire on sleep behavior in the patients. And hopefully we can correlate some of these findings with what we see in the human population and help come up with endpoints that we could potentially use in a clinical trial for a drug. And yeah, that's all I wanted to talk about today. It's one of my side projects for my PhD research. This is the Gray Lab, and you'll hear from Steve later on. And yeah, thanks for the DDX3X Foundation for being so supportive. And I think we can move on to Jen's talk.
Yep. Up next for our fourth is Jen San Miguel from Jackson Labs, who's joining us remotely. So I'm going to try to bring up her little picture here. All right, go for it.
All right. Hi, everybody. My name is Jen San Miguel. It's so nice to see you virtually. I am a PhD trained scientist. I work at the Rare Disease Translational Center at the Jackson Laboratory. I wanted to start out by just explaining who we are and what we do and how we became involved with the DDX3X Foundation. I'll move this out of the way. So our center is really geared towards empowering rare disease solutions, and we do that by partnering with foundations, academics, clinicians, biopharma, to develop models, test new therapeutics, and really accelerate that journey from diagnosis to the clinic. So that's our overall mission. The way we approach this is slightly different from maybe an academic lab or a pharmaceutical company. We're very patient-centric and family-centric. So what happens is we are approached by whatever organization, in this case, it was the DDX3X Foundation and a number of the academic researchers who are in the room with you today. And we discuss what are the patient variants that we know about the disease. Are there any mouse models that exist? And we take this into account when we think about, are there new mouse models that we need to make? We think about what is the mechanism of the disease, and we know that depending on what type of variant the different patients have, this might change even within a particular gene, but we try to think about if there's a particular variant that we're interested in modeling in a mouse, what would the therapeutic look like for that variant? And then we engineer the mouse model if there's not one that exists, or we can also work with various cell lines, including induced pluripotent stem cells, mouse embryonic stem cells, which I'll mention in a minute, or immortalized cell lines, so things that are just kind of floating around, like cancer cell lines that we can use for basic research. And then the idea is if we get a mouse model, we want to phenotype that mouse, which means we're going to look at things like you already heard about, anxiety-like behaviors, motor function, sleep, to understand if our mouse looks like what the human patients look like. And if so, then we take the therapeutic that we already have pre-thought about and either partnered with somebody or started to develop in-house and put those two things together. And this starts that preclinical pipeline that is essential for moving things forward into the clinic. And so we do this across our program. I think we have over 65 different disease areas that we have within our center, and DDX3X is one of those. So we were approached, and it was decided with the group of investigators that we would make a mouse model that carried the R488H variant in DDX3X. And this is because a number of different variants were already being engineered across different academic institutions. And there was interest in this particular variant due to some different cardiac features that were being observed in these folks. So let's see if I can advance my slide. There we go. So our project goal is to develop a mouse with this variant and understand if it looked like the people who carry this variant have. So at a very high level, We knew we couldn't engineer in that single DNA change into the mouse gene due to the way that these variants are transmitted from parent to offspring. And so we had to do something called a conditional approach, which is where we engineer the DNA in a way where we can turn the mutation on. So what I'm showing you here is a gene structure of the back end of the DDX3X gene. You can see the exons are these boxes, and those are the pieces of DNA that actually code for the protein. And then there are these flat lines that are just intervening sequence called introns that don't end up getting translated into protein. And so what we did is we engineered this little piece of DNA that's it's called floxed. It has these little pieces of DNA that can be recognized by an enzyme that can actually cut this piece of DNA out. So our mouse would translate the normal or transcribe the normal DDX3X gene. And only when we expose it to this protein called a Cre recombinase, this chunk of DNA gets cut out. And then we have the exon that carries a single DNA change that would lead to the R488H change in the protein. So we're basically going from a wild type mouse to a mouse that would have this variant. And this allows us to bypass the important embryonic expression timing of DDX3X. So we can actually get a mouse that expresses the variant and allow us to study the biology in this mouse. Unfortunately, what we noticed pretty early on is that our DDX wild type mice weren't actually wild type. So the number of animals that we were getting out of the crosses was low. The breeding efficiency, like how many pups there were per litter, wasn't making sense. And so one of the things we did to investigate this was we looked at the actual protein itself. So this is called a Western blot. What we do is we take all of the protein from a particular tissue. In this case, it was the brain. And these are all plus mice. These are all actual wild type mice that have no engineered DNA at all. And we can use an antibody to detect the DDX3X protein. So we actually label it and we can see it as a band on a gel. And the numbers indicate the size of the protein. So here you can see the DDX3X protein in wild type mice. And then in our engineered mice that were supposed to be wild type, you can see the bands are a lot lighter. This means there's less protein. And then we actually have this weird band up here that's not supposed to exist at all. And this was because there was a defect in how the gene was getting processed into protein. And that defect is something that we can actually work with. So we pivoted away from actually physically using mice in this case, and we went back to the drawing board and started working back in cells. And the approach here was to take the sequence that was causing problems in the way the gene was being expressed. We are actually able to use CRISPR-Cas9 technology, which some of you may have heard about, which can change the DNA sequence. And in this case, we were changing a sequence away from this AGGT combination that was leading to problems and changing it to GCC, which keeps the protein exactly the same, but just changes the DNA sequence and should, in theory, fix the way the gene is getting put together. So we are using two different guide RNAs. They match up to the DNA sequence and allow CRISPR to cut and change the sequence that we want using a piece of DNA that we introduce into the cells. So basically like a template that the DNA can copy itself onto to get this corrected sequence. And so we've done a number of things so far. We've been able to correctly target at least a number of different cells using this strategy. So that's the DNA checkbox right here. And what we're currently doing is understanding if that DNA change we made in the cell lines is actually leading to the proper RNA sequence, which would then turn into the proper protein. So that's where we are today. This gene is, I think, particularly tricky to manipulate, but that doesn't mean it's insurmountable, and I think we're making a lot of progress. So thank you for your time. This is our center. We have over, I think, 75 people now in our group. So if you're ever in Bar Harbor, Maine, please come visit us. Thank you so much, and happy to take any questions.
Okay. So thanks to all of our presenters. Do you want to start us off in the back with a question? Chelsea, could you grab the mic and pass it in the back there, please? Thank you. So we can take questions about anything that you've heard about or other stuff that you haven't heard about.
This question is for Sylvia. You had mentioned that there were motor declines that you were seen in the mice later in life. And I was wondering how much later in life did the mice experience those motor declines, and how does that translate for humans?
Thank you for that question. So let me first take the first part. So years ago, we first aimed at looking at the longevity, right? How long do they survive, these mice? And mice in captivity can typically survive up to 2 years of age. And our DDX3X mutants do survive up to 2 years of age. So we do not see change in, the lifespan. So we aimed at characterizing these 2 years old mice, but then COVID hit, so we could not characterize the 2 years old. But we had staggered a year and a half and a year prior other cohorts that we could look at likely. So the deficits, we see them in 1-year-old mice, which in human age would, again, this is, mouse and humans, but it would be around 50. And the reason why we ran that experiment is because there was one single report in a study from Baylor, I think, that showed that there was a regression in a 47-year-old woman, right? A motor regression. So we saw the motor decline in the 1-year-old mice. And then as I was presenting these initial data at a DDX3X conference, I want to say in San Antonio, Texas, there was a father in the audience who stood up and asked me a very, smart question. And he said, but Sylvia, your mice doing all the therapy our mice do. And I said, that's a fair point. I'm telling you that there is a motor decline, at 1 year of age, but those 1-year-old mice have not gone through anything. They've spent their life in the cage, as Andrea showed. So I said, okay, so we're gonna go back and test a separate cohort of animals that had gone through a lot of behavioral training and testing in adulthood, and the motor decline was no longer there. So showing, and this is published work that I've presented in the past. So this shows that, again, that motor trajectories is malleable, is still plastic even in adulthood. And this is the piece of finding that inspired Lauren to look even earlier, right? So we had done the motor training and in sort of testing up in adulthood. Can we do that even earlier in juvenile age and prevent entirely the motor deficits? Does that address your question? And then for the human part, the answer is that we do not know, right? There is that single case that is what inspired our initial investigation, but it's not that we have a characterization of the adult population.
Thank you for that. Any other questions? Over here. One in the back. So the question was about connecting sleep observations that people have made with your clinician and potential pharmacology and treatments.
Yeah, so sleep definitely has a lot of connections with other behaviors, like you mentioned. And one of the things that we saw in the mouse was this like reduced time spent in paradoxical sleep and the REM sleep, and that has a lot of known functions with memory consolidation and emotional regulation, right? So there, hopefully, with some of this data, it can help inform the natural history study with what questions they're asking specifically for the DDX3X patient population in that study. For clinicians that aren't using those measures in the that are in the natural history study, it's definitely more challenging. There's a few published work on kind of DDX3X presentation and clinical care suggestions, and that has a few, I think there's a sentence in there about sleep, like one sentence in there about sleep. And it's because it hasn't really been looked at in the patient population. And it is one of the reasons why I was interested in looking at it in the mice, because I was hoping that, like, there's a few ways research can work. You can either have clinical data and then like, I'm going to go back into the basic research lab and try to find that finding, or it can stem from the basic research lab and then re-inform clinicians on what they should be looking at in patients. So it's kind of a back and forth, and it's something that we are doing a lot right now, is talking with the clinicians and presenting this work from the basic side so we can help kind of fully understand the clinical picture. But yeah, we definitely know that there's not much published in the DDX3X population on sleep, and it's one of the reasons why the natural history study is going to be assessing sleep, and it's one of the reasons why we were studying it in the lab, to try to help with that.
Yeah, I also want to plug there, the foundation and the registry, right? Because, you all may have observations about your own folks, right? That then if we collect that as a group, we can use that actually to connect even with the natural history study and say, oh, you're measuring this thing here, but maybe you should also be measuring something else. And the best way to get that information is to understand that, 20% of people are having this particular thing that we're not studying at all right now. And that can feed then back, both into clinical care and also into research to motivate people to look at it. Anything else?
Well, thank you for your presentations. One of the questions I had is, at the very first presentation, we talked about a split between missense and nonsense variants. And I was wondering, in the studies with the mice that we've done, are those, there was a percentage of like 51 to 49%. Is that also the split of variants that we try to do in the mouse models, or is each mouse model specifically tied to a specific variant?
Yeah, thanks for your question. If money were endless and time were endless, we would make mice for all of the different types of mutations. So we can't do that. But what we can do is, for the nonsense mutations, those tend to all give a similar impact. We make the assumption, based on a lot of molecular research that has been done, that the nonsense are going to be reduced levels of DDX3X. And so we can use one mouse model, essentially, to understand that. And so, and that allows us to model about 50% of the affected population. So in terms of the missense is where it gets more challenging, right? So we have engineered, using similar approaches to what we heard about from the last talk, conditional mice that allow us to express the T532M mutation. We selected that one because, as many of you know, sometimes you will identify someone in this room whose child has the same exact variant. And so we reasoned that by focusing on variants that were in common across several individuals, it could give us the most chance to make an impact and to start to learn about trends. So that particular mutation has 5 variants in common across that have been identified in 5 different individuals. There's, I'm sure, more out there. But the goal of this really was to be able to do comparisons and just at a high level understand how different classes of mutations behave. And so that's how we've approached that.
I'll add maybe just a wrinkle to that, which is that, in girls that have 2 X chromosomes, they have 2 copies of DDX3X. And one of the things that is unique about this gene is that both of those are made into protein. Usually with the X chromosome, you block production of 1 copy so that you just have 1. And then in men that have an X and a Y, you still just have 1. And so when you have the missense mutations in particular, we don't always know how much those are being made into protein. And sometimes they may end up looking more like a loss of function or a nonsense mutation, even though they're actually a missense mutation because the cell has shut down the DNA. So studying those mutations is super important. It's definitely something that we should do. But even when you see the same mutation in a person, you might meet all 5 of those people that don't have exactly the same presentation. And that might be because they're making slightly different amounts of protein. So, it's a great starting point, and it's definitely something that we should do is study the missense mutations, but we also want to know that, people are not mice and people are very complicated, and, having the same mutation doesn't mean you're going to be exactly the same as another person. We have time for 1 or 2 more. Okay, in that case, let's thank the panel and we'll move on to gene therapy. Okay, so for the next section, we're going to have a presentation by Stephen Gray, who's focused on gene therapy. You may have heard about many different flavors of gene therapy. What we're going to be talking about primarily today is the idea of replacing the We're talking about really replacing with another copy of the normal DDX3X gene, which I think is especially effective for people that have these nonsense or splice site variants. There's other strategies we might be able to use for people with missense variants. And Stephen's an expert in this space. He's done a lot of work in many rare disorders by using viruses to restore expression of a DNA. And he's going to tell us about some of that work with DDX3X today. And so we'll have a presentation and again, time at the end for questions.
All right. Pleasure to be here again. Good to see some people that I know well and see a lot of new people. So Andrea is a graduate student in my lab. We've been working on DDX3X gene therapy approaches for about 5 years. And so what I want to try to cover here is give you just an overview of gene therapy in general, sort of what it's starting to be applied to, and then go into what we're doing specifically with DDX3X and what the next steps are. And I'm coming from UT Southwestern Medical Center in Dallas. All right, so just again, start everybody on the same page, DNA, RNA, protein, and I wanted to talk first about some of these different mutations and what they cause, and to try to explain this a little bit. So this is, your DNA code is made up of these 4 letters, A, T, C, and G, and they're strung out, and there's about 3 billion of them that make up all of your DNA. And your cells have machinery that they're able to like scan across the 3 billion letters and pick out sentences. And there's different signals to tell them where those sentences are that confer information. And so this would be an example of like a sentence that your cell would see. Now, to you and I, it doesn't really make much sense, but your cell can read this. So just conceptually, I'm going to convert this to a sentence that we can read. And I presented this last year, so some of you may be familiar with this, but so to make this easy, I'm putting it into a string of 3-letter words, and this is when your cells read these sentences, it breaks up that string of letters into little 3-word or 3-letter words. So in this case, I've converted to the cat ate the rat and got fat, the end. Okay, so it's able to see the start of that sentence, the end of that sentence. Sentence makes sense, sentence tells a story. So we would call this like the normal version of the gene, or what scientists will call like the wild type version of the gene. And then you can have changes, single letter changes. The cat ate the bat and got fat, the end. Okay, so sentence still makes sense. It has a little bit different meaning. So this would be a missense mutation, and we might call this a variant of unknown significance, or a VUS. So it's a change, but we don't know if it actually causes disease. But then you could have something else, same single letter change in the same location, the cat ate the eat and got fat, the end. Okay, so again, you can see the sentence, but it doesn't quite make sense anymore because of just that 1-letter change. So this is a missense mutation, and this might be pathogenic. Maybe it's mildly pathogenic. it causes a disease. Disease isn't really severe because most of the sentence is intact, but it just doesn't quite make sense. But then you can have things like a premature stop. The cat ate the rat, end. So again, single letter change, but now the sentence, we're missing information. So this could be this could still have some partial function because like the sentence still does make sense, but you're missing half of the information. So again, this could be like pathogenic or it could be mildly pathogenic, all right? But then you can have things like a single letter deletion or multiple letter deletion, and your cell reads these words in order, 3 letters at a time. So if you're missing 1 letter or 2 letters or 4 letters, something else that's not a multiple of 3, then it shifts the frame. So now it's going to read these letters in order. And so after that small deletion, now the rest of the sentence doesn't make sense. So this would be a very bad mutation. This would probably be complete loss of function. And then you can have the same thing where you have like a letter or multiple letters inserted. Exactly the same effect. It shifts the frame, and now the sentence doesn't make sense. Okay? So all of these mutations that I've covered so far, even though they're all different, a frame shift, a premature stop, a missense, the result of all of these is that we have reduced function. We've lost expression. So even though these are all different types of mutations, they could all result in basically the same thing. It's less DDX3X, less functional DDX3X. So the outcome of all of these, even though they're different mutations, the outcome and the impact on the patients can be the same. Now we can have something weird that happens sometimes, and it looks like it probably happens with DDX3X, is you can have a missense mutation where it changes a letter, and now you have a new meaning. So the cat ate the rat and got fit, the end. So instead of the cat getting fat, now the cat is getting fit. Okay? Sentence still makes total sense, but the meaning is different here. The meaning is kind of opposite. So this would be, now we've made a new function for the protein. And in this case, I think this is probably why some of these missense mutations that the girls seem like they're maybe more severe than some of these other changes where we're just losing DDX3X. Okay? So I hope that kind of illustrates what these different mutation types are and how they really, this is how they translate into, the effects. All right, so now into gene therapy. So this is a book that was published about 15 years ago, and they coined this term called the forever fix. And this is the aspirational dream of gene therapy, that we can go in, we can fix the DNA, it's a one-time treatment, and it's a kind of forever lifelong fix. That's the hope, that's the dream, because we're going in and trying to fix the DNA. And once you fix the DNA, then that's permanent in the cells. All right, so how does this work? You guys are very familiar with this first part where patient has a condition, you're going through some kind of diagnostic odyssey and identify that the patient is the way that they are because there's a defective gene, right? You know this really well. So the idea here is to package a working copy of the gene into some carrier like an engineered virus, use that virus to deliver the gene back into the body. Now, again, we're using that to deliver it and put it in permanently. So the virus is not replicating, it's not spreading, it's literally acting as like a little molecular mail truck to deliver the gene and then go away. So right now, the gold standard, the tool that's being used to deliver genes to the brain is something called AAV. It's adeno-associated virus. It's not adenovirus. It is actually in the parvovirus family, but it is has a distinction of a human virus that doesn't cause any disease in humans. It's kind of a parasite on other viruses. So it's inherently has some kind of safety features where it doesn't cause a disease. And again, we're gutting this virus. We're taking out all the virus genes and sort of tricking it to package some therapeutic gene we wanted to carry. So it doesn't replicate, it doesn't spread, it's not going to cause a zombie apocalypse. And we can make modifications to the shell of the virus to target it to specific tissues and specific cells. So this is being used in over 300 different clinical trials for various diseases. And now we have several FDA-approved gene therapy treatments that are marketed, that are prescribed by doctors. Going back to, I think, 2017 was when the first one came out. Okay, so way to get here are the ways to get AAV carrying genes to the brain. When I started in this field about 20 years ago, if you wanted to deliver a gene, deliver AAV to the brain, you drilled a hole in the skull, you stuck a needle into the brain, and you injected the virus. So that's been done safely. There's one FDA-approved product for something called AADC deficiency that does that. But in the case of something like DDX3X, where you have to inject, or you have to get the gene spread all throughout the entire brain, you can't do enough injections. Like you can do it in a mouse, you can't really do it in a human. So one of the things that I was very involved with early on, starting about 15 years ago, is looking at different versions of AAV that we could deliver to the brain different ways and get more broad spread. So there's one FDA-approved product for something called spinal muscular atrophy that uses an AAV that's injected IV, and then a small part of that virus gets across the blood-brain barrier and into the brain. It's a very, very effective treatment for that disease. So that's one approach. But there are a lot of problems with doing that with the current AAV technology. So what we've done and what most of the field has done is shift to basically administering this by a spinal tap. So your spinal fluid is actually produced in the middle of your brain. It flows down your spine, it flows up and then around your brain. It's one continuous fluid that kind of bathes your entire brain and spinal cord. So you can inject drugs like AAV into that fluid, and then it'll spread. Okay. So you can do that in a mouse. We do spinal taps in mice, believe it or not. And if you do this in a mouse, this is a cross section of a mouse brain, and all the little black dots that you see there are cells that are expressing the gene. So there's a couple things to notice here. We're seeing spread throughout the whole brain. We're getting the gene delivered throughout the whole brain, and this is permanent. You'll also notice that the whole brain isn't black. So it's not every cell. In fact, it's probably around 10 or 20% of cells. And so the current technology with AAV, this is about as good as we can get. But, you know, that's in a mouse. We can also do this in monkeys. And this is just, let's see, the one panel up there in the upper left is slabs of a monkey brain cut through from front to back. Okay. And then just highlighted one of those that we, just to illustrate this, where we processed it to slides. And then these little red dots here are staining for our gene being expressed. So again, you see that it's sort of spread all throughout the brain tissue. We're getting gene expression in all the different regions of the brain. But again, it's a subset of the cells. It's not every cell. Okay, and so this is important, and I'll come back to this here, where This is good, but it's very far from perfect. But this has been applied pretty extensively. This isn't a comprehensive list, but this is a review that we did a couple years ago of different diseases that are being treated with this approach, like using AAV, injecting it into the spinal fluid, and delivering broadly across the brain. And I'll highlight there are also, this approach is being used for 2 gene therapy trials for Rett syndrome, and I'll touch on this in a minute because it's pretty relevant. Okay, so some practical limitations of this. Again, as I've highlighted, the current technology can't deliver the gene to every cell in the brain. It's a minority of cells. So that's the current technology. So think treatment, don't think cure. Let's be realistic. Genes are being delivered as kind of mini chromosomes, so they won't be regulated the same way that DDX3X is in the body. So the treated neurons might express less DDX3X than normal. So this can be beneficial, but not like 100% back to normal. Treated neurons might express more DDX3X than is normal. And this could make things worse. So we've got to be really, really careful here that we kind of fine tune this and design it properly. Okay, so again, think treatment, not cure. Okay, the other thing is this is, as I said, the hope and the dream of gene therapy is it's a one-time treatment, lifetime benefit. That's the great thing. It's also the scary thing. You get one shot and then it's permanent, for good or bad, it's permanent. So after it's delivered, the DNA is in the cells permanently, the gene is being expressed, and we can't turn it off. So if we get this wrong, we can't turn it off, okay? So this isn't something where you can just stop taking the pill. So I'm just, trying to be real with all of this, and why some of these things take a while, because we have to make sure that we really get this right. Okay, and so by the numbers, and I did this calculation a couple years ago, and it kind of blew my mind a little bit because we just start making AAV and dosing it and everything else. But the average number of cells in an adult human body is around 30 trillion. For reference, if you have a COVID-19 infection, you'll end up with about 100 billion COVID-19 virus particles in your body. When the trials that we're doing right now, like that we're doing, injecting AAV and delivering it to the brain, we're injecting about 1 quadrillion engineered AAV particles into the spinal fluid in about 2 teaspoons. So that viral load is 10,000 times more than what you would have in your body during a COVID-19 infection. So, I mean, these are big numbers, and there are implications of this. And that's one other thing to expect, is that if you're doing gene therapy, there is a degree of immunosuppression involved, because if you've got all this virus load circulating around your body, your immune system will freak out. It won't be very happy with this. So we usually incorporate, or we always incorporate, some transient mild immunosuppression, just to tell your immune system, like, just ignore what's going on, it'll go away. And that's been done very effectively and very safely. And this high virus load that's in the body, it's transient. It starts to clear out within 24 hours. All right. So all that said, all the good and the bad and everything else, we've been, just my lab alone has been fairly prolific in developing this technology and then applying it to different diseases. And this is not everything that my lab works on, but this is just an illustration here of the ones on the right are things that we've developed and we've moved into clinical trials. So these are all dosing in patients. Going back, the first one was in 2015. So we got about 10 years of experience with this now, using this exact delivery approach. And then IND enabling in the middle, IND is an investigational new drug application to the FDA. So these are things that we're actively working right now to move into a clinical trial in the next year or two. We're going through toxicology studies, we're going through drug manufacture, everything else. And then the discovery is the things that are right now in the lab that we're still trying to optimize. So there's DDX3X. And I'm also going to highlight a couple slides here on Rett syndrome because of all these different diseases. This is the one that is, probably most analogous to DDX3X. We started working on Rett syndrome in 2007, before all this technology was mature. And so the background on Rett syndrome, this is another X-linked disorder. It's found in girls. It's typically fatal in boys. And this results in severe neuromuscular defects, severe developmental delays, intellectual disability, breathing problems, seizures. This is actually more severe than DDX3X. Similar to DDX3X, it's caused by de novo mutations in this gene. So it's not necessarily passed on from parent to child. This is just a spontaneous mutation that happens. And the gene here is called MECP2. And what MECP2 does, just like DDX3X, is kind of a master regulator of other genes. Same thing with MECP2. It regulates the expression of literally thousands of other genes. So you have all these downstream problems that happen. Let's see. And some of the complications with this, with Rett syndrome, is we know that if you're missing MECP2, it's bad, you have Rett syndrome, but there's another disease where if you have an extra copy of this gene, it's equally bad. So the gene itself is very dose sensitive. And so it took us a while, but we kept working on this, and eventually we found a way to get this right. And so this technology got licensed to a company called Taysha Gene Therapies. They took it out of our lab and moved it into patients. And so this is going back to about 2.5 years ago in 2023, where, and let me back up. So Rett syndrome, girls will develop normally until about 18 months old. Then they can stall, they can regress. And after about 6 years old, they never acquire new skills. They never pass new milestones after about 6 years old. When Taysha started this gene therapy trial, the first patients they dosed were adults. Some of that was for safety reasons. And so this is just a press release that they issued after the first patient was dosed, that showed him, in this patient, 4 weeks post-injection, started showing improvement in multiple domains. For example, improvements in breathing patterns, vocalization, motor skills. Patient was able to sit unassisted for the first time in over a decade. She demonstrated, and Rett girls will have kind of really severe hand clasping and stereotypies. So she was able to unclasp her hands and hold an object steadily for the first time since infancy. Now, Taysha's put out various press releases after this, and this is their most recent from earlier this month. They presented some of the longer-term data across a cohort of 10 patients that have received this, some adults, some children, children as young as 6 years old. And so this is just a snapshot of a summary from a poster that they just presented. And all 10 of the patients showed improvements in terms of developmental milestones or other skills and improvements. So they didn't go from being nonverbal and non-ambulatory to, running a race and reciting poetry, but there were examples of patients being nonverbal and going to using phrases and sentences with meaning. going from understanding simple words to understanding complex ideas and engaging in conversations, about being able to make choices less than 10% of the time based on eye gaze, to being able to make choices 100% of the time by pointing, fine motor improvements, going from like severe stereotypies to highly reduced stereotypies, going from no purposeful hand use to playing with toys, transferring things intentionally, requiring caregiver-assisted feeding to being able to self-feed and hold a bottle. Let's see, gross motor improvements. I'm just reading through these, but going from severe dystonia to no dystonia, non-ambulatory to being able to walk with support, requiring caregiver support to stand, being able to pull themselves to a standing position, reductions in seizures, going from requiring a G-tube to oral feeding. So, this has been, for me, this is out of my hands at this point, and I'm an observer, but it's been pretty amazing to watch. And keep in mind, this is with this delivery approach that I explained, where we're still targeting a minority of cells across the brain, and we're able to see these benefits. So going on to DDX3X and where we are with this, so the project in my lab started in 2020. This was actually originally funded by Taysha Gene Therapies, but a few years ago, there was kind of a downturn in the biotech and gene therapy market, and all companies were like dropping different things, so Taysha dropped this. The foundation stepped in and picked this up, has been funding my lab from 2022 to 2026. And so I'm directing this with somebody else in my lab named Xin Chen, and then Andrea and Rachel are really the boots on the ground doing the day-to-day stuff, and they're here. Okay, so start with the bad news. We took an initial push on this using the delivery approach that I'm talking about. Oh, hold on. My slide's not advancing. Should I try to reshare? Oh, hold on. I'm reconnecting to Zoom. Oh, sorry, I won't be able to share this with you. No. Yeah, all right, hold on. Let me try to rejoin here. Wow. Sorry, it looks like the hotel kicked me off of Wi-Fi.
Okay, I think.
Well,
Yeah, I'm a chin.
all right, I think we're almost back in business here. They're just gonna let me back in, and then should be good to go. All right, I think I'm back in business. All right. All right, so I'll tell you where we are with DDX3X. So we had an initial design of this. All right, we had an initial design of the, how we're expressing DDX3X in the AAV. We delivered this to mice, and we saw some really modest improvements in the mice, like this anxiety-like behavior that Andrea was talking about. We improved it a little bit. It wasn't that impressive. But when we treated normal mice that don't have DDX3X, we did that too. We actually induced kind of anxiety-like behavior. So we basically, and then if we injected this really by a spinal tap, really around the spinal cord, then we induced like a movement disorder in the mice. So this showed us that, there was a lot of evidence that DDX3X can be toxic if we provide too much of it. We kind of suspected that might be the case, but this kind of proved that. And just to hit that home, we designed a different version of this where we included some elements in that would just express like lots and lots of DDX3X, like really drive high levels of DDX3X expression. And when we injected that into the mice, they all died within a week. So we know, just like our, what we had experienced with Rett syndrome, that DDX3X, we need to be really careful, that we need to have mechanisms to kind of fine tune the expression. We can't express too much. We need to be really careful here. And like I said, this was exactly the situation we encountered with Rett syndrome. We did get through it, and we have a treatment in humans that seems to be working pretty well. But that means that DDX3X is what we'd call a Goldilocks gene. It can't be too strong. It can't be too weak. It has to be just right. So it is possible to design this, and we've had success doing this. But it is another level of difficulty, and it takes time. And that's where we are right now. Now, we have two things that are going for us here as we move forward. One is we've been working with this version of AAV, AAV9. That's what we used in the Rett trial. That's what we've used in everything that we're doing. There's newer technology. There's been like next generation versions of AAV that have been created. This is coming from a lab at MIT that we're collaborating with. We've got this version of AAV, and basically they injected AAV9 and this next generation version into mice at a relatively low dose. And so the bottom is AAV9. That bottom picture is AAV9. That's what we're using right now. At that low dose, it is actually expressing in the brain. The purple is showing the gene expression, but it's just really low. So you don't really see it here. But this is their new advanced version of AAV that I think you can see a night and day difference. When they quantify it, it's about 50 times more potent. So, and this is moving away from this spinal tap approach, and it goes an IV approach. So it's a one IV injection, and the advantage of that, when we inject the virus into the spinal fluid, then there's areas close to the injection site that kind of get a lot of the gene, and then there's areas away from the injection site that get less of the gene. So it's like a gradient. In this approach, since we're putting it into the bloodstream, it's basically accessing the entire brain and spinal cord all to the same extent, so you get much more even gene delivery. You don't get one cell that has 100 copies of the gene and one cell that gets one copy of the gene. So it's much more even distribution, which if we're dealing with a dose-sensitive gene, that's really important. And then also because it's just so much more efficient, we can go in with lower doses. Okay? The other advancement, and this is all, these are advancements that were funded directly by the DDX3X Foundation that, we've been working on trying to build in a regulator to control the expression of DDX3X. And we did something similar. This is how we solved the Rett syndrome. And in this case, we went from an expression, sort of this mini chromosome that would not have that regulator. That's what we were using before. We identified a natural element that DDX3X normally uses that basically, if you go into an environment with low DDX3X, which are your girls, then it allows high activity of this gene therapy. If we go into an environment with normal DDX3X, then it would lower the activity of this. So it actually, it provides some buffering capacity, pushing the expression up or down based on whether there's a need for DDX3X or not. So this was a good breakthrough. And right now, kind of combining these two things of the much better delivery technology and now our ability to provide some level of buffering and regulation, then we're kind of poised to take another good push with this. There's still a lot that can go wrong, but I'm optimistic. So looking beyond that, just for everybody's knowledge, this would be like the roadmap of like getting a drug into patients. This is really for any drug. We're kind of at this proof of concept stage where we're trying to treat mice. We know how to do this. We know how to do this in a way that the FDA get the kind of data that they're looking for so we can make a hard push. If that's successful, then the next steps would be talking to the FDA and then starting to work with a manufacturer to produce drug for a toxicology study, do these formal toxicology studies, then manufacture like the human drug, and all of that goes into an application to the FDA to start a clinical trial. And the clinical trial design is very, very informed by the natural history study and the registry. Those are really important. And I'll just put sort of some ballpark timelines and costs associated with this. It's a multi-million-dollar, multi-year endeavor to take something from mice into humans. And the landscape of this in the bigger picture is, you kind of have these ramp ups in cost and an amount of work where we're at this beginning stage of trying to show that something's safe and effective in mice. And then once you go into initial human testing, like to get through a small human clinical trial is, again, I'm put ballpark like $4 to $8 million. And then if you're trying to like go from treating a handful of patients in an initial clinical trial to an FDA-approved drug that all patients have access to, then you're in the tens or hundreds of millions of dollars. Big pharma will say it's $2 billion. But for a rare disease like this, there could be better. So, some of these initial steps can be funded through like private foundations, grants, philanthropy. But when you start getting into like developing something to a commercial product, it's just, you have to bring in some kind of biotech or pharma. So I hope that this has been helpful. I'm here to answer lots of questions. And thanks for listening. Yep. Yeah, so at this stage, like once the clinical trial open, then it goes on clinicaltrials.gov. Then there's different sites, actually around the world that are recruiting patients. And then they have a certain number of patients that they recruit. Once that's done, then there's a pause. And like right now, they're moving into a phase 3 clinical trial, which is supposed to be the pivotal trial to lead to an FDA-approved drug. So right now, there's just like a limited number of spots. It's experimental. And when you're participating in a clinical trial, then there's the hope of benefit, but there's also risk. These are really courageous, brave individuals that are volunteering to test this experimental therapy. Yeah.
Move forward with stuff. I saw the talk about putting the regulator into a potential treatment to help with some of that. Would your vision be then there's one treatment, or would it still have to be like an individualized treatment based on the individual being treated? Like, obviously we have the nonsense, the missense, frame shift. I love your slide, by the way. I still have the picture from last year. It's the only one that's ever made sense to me on that. But just thinking through that differently, and obviously I'm in the missense side, so my brain goes to that directly, and we know how different that is. Are you thinking it would have to be each patient getting an individualized treatment, or it's one that would work for just about everybody?
Yeah, I mean, we're trying to design this as it's just a way to introduce more DDX3X into the system. That's our approach. So we're really treating a DDX3X deficiency. Many mutations would lead to a DDX3X deficiency. So our hope is that this would be a pan therapy that could apply to everybody. But there are some of these missense mutations that are like a toxic gain of function, or they're thought to be doing that. And so it's possible, the short answer is I don't know. In Rett syndrome, you kind of have the same situation to some extent, and right now it's being tested in everybody. But it's possible that a small set of the missense mutations might be incompatible with this, or maybe it's just less effective, but it may not. It may be fine. But right now we're testing everything as it's a DDX3X deficiency, and we're just trying to add more into the system.
And I just want to add on that this is one flavor of gene therapy. There's also other flavors of gene therapy. You'll see on your schedule that I'm supposed to be Sarah DeSalvatore, which I'm obviously not, who's a fantastic DDX3X mom that couldn't be here because of a family situation unrelated to DDX3X. She actually works at a gene therapy company that uses a very different strategy to kind of stitch together a synthetic RNA with the mRNA that's made from a gene. And that might work for different classes of mutations than what Stephen is talking about, right? So this is really kind of the name of the game, is to take, all the different research strategies that you could have for gene therapies and match it up with patients where it's most likely to have a safe and, impactful effect on them, right? And that takes all the science that we've been talking about and also the clinical understanding.
Yeah, and look, I'll add to that. There's also gene editing, which is a type of gene therapy, and there has not been a gene editing treatment that has actually moved into clinical trials for the brain. There are efforts that are trying to do that, but gene editing would be very targeted against each specific mutation. And so this would be an idea of having one treatment that could apply to everybody, versus like a gene editing would be taking the rare DDX3X disease and dividing it up into like a couple hundred ultra rare forms. And each one might be like a multi-million-dollar effort. So it's kind of not really, it maybe could be done, but it's not very practical. Yeah, so our approach is you have the mutated version of DDX3X that's in the patient. We're not touching that. We're just leaving it alone. And we're adding a new copy of the gene kind of on top of it. Gene editing would go in and actually physically correct the patient's DNA. Yeah. And the advantage of that is then you're leaving all of the regulation systems in place, and it's perfect. So the hope and the dream is that would be like the way to go, but the reality is there's a lot of challenges to doing that. Yeah, well, so Beth is asking like what? Beth is asking why I feel optimistic about this. So it's basically that it's these two advancements. So one is this newer technology, like these newer versions of AAV, are really like they're serious game changers. They're really set to revolutionize the whole field of like gene therapy for the brain. It's just making everything a lot easier, from being able to like barely deliver the gene enough to now, with this, the data is like they're showing gene delivery to like over 80% of the neurons across the brain. Like it looks really good at lower doses than what we're doing now. So that's just a tool that makes our job a lot easier. And then, again, the gene regulation strategy, the first time that we went in, we didn't have any mechanisms in there to kind of control the levels of DDX3X after it went in. And now we've identified some elements that naturally regulate DDX3X that we've shrunk down small enough to fit into our virus. So it's those two things coming together that make me feel pretty optimistic. And also just I've done enough things, all of our work with Rett syndrome and other diseases where we've dealt with dose sensitive genes. There's still plenty that can go wrong, but we've got some good tools and good approaches that this is reasonably likely. Yep.
First, I just want to say thanks for dedicating a lot of time and effort to this for children that we will maybe never have a personal connection to. As a mom, it means a lot. I love the Goldilocks analogy, so I guess I'll try to play off that to ask my question. Based off the variability in this, it seems like there are maybe some kids that have very hot porridge like Papa Bear, and then some very cold porridge. And so we're wanting the baby bear, just right porridge. So with the gene therapy, is it able, if I'm understanding this correctly, it would be able to go in and to decide like, okay, this is hot porridge, so we want to bring it down a little bit. This is cold porridge, we want to bring it back. Is there like a certain like number, or I don't even know, like expression of the DDX3X gene that you're kind of looking for? Like, how is that determined? I don't know if my question even makes sense.
No, it makes total sense. So the experiments that like Rachel specifically has done in the lab is basically have like this regulated DDX3X, and we'll put it into cells where we've deliberately like knocked down the cell's DDX3X. And then we see that the gene that we put in is stronger, it's expressing more. And then we have cells where we put it into cells where we've deliberately overexpressed DDX3X. And then it kind of like shuts it down, so it's not expressing as much. So again, it's sort of, we could over deliver it and we could break through that, but we're trying to target kind of the natural level of DDX3X expression. And at least now, before we would just put it in and expresses whatever it expresses. But this time we have something that'll at least adjust to how much DDX3X is in the cell. I hope maybe that makes sense.
Yeah, that does. And if I might ask one more question that as well, and I don't know if this is more like a targeted gene therapy as opposed to this seems more like multifaceted, all the different cells. But for a kid, let's say with DDX3X, it has more of like a motor development versus maybe a speech development. Is there a way that you see that this gene therapy, maybe Rett, and how you've seen it expressed with Rett syndrome specifically, like where you're able to kind of see an enhancement, like, hey, this child really struggles motor-wise, or as, hand stairgazing you're referring to with Rett, that you're able to kind of specifically target that type of area?
I mean, it's really just across the board. In terms of a rescue or a benefit that we might expect, I don't want to comment too much on that because it's really forward-looking. We're going to be trying to restore at least close to normal DDX3X expression in lots of cells, probably the majority of cells across the brain. And then whatever correction happens after that is going to be really dependent on like the developmental age of the patient and what capacity the brain has to kind of relearn and rewire. There's a lot of factors there, and we can model some of that in mice. And there's a lot of that we really won't know until we go into patients, right?
Thank you for your time.
And that's the case with Rett syndrome. We knew in the mice that the earlier we treated them, the better it worked. So they went into adult patients not really knowing if it would help, and it did.
Maybe one more.
Thank you. Just to piggyback off of her question, I thought from going to conferences in the past, I had learned that with mice and probably with our kids, that they don't have as many like, I'm going to say it wrong because I don't have a science background, but like neural synapses, or they're not making as many neural connections. So does that same issue present with Rett syndrome and the children that have been treated with the AAV, where they're seeing improvement, have they noticed that there's more neural connections and that's the reason for the improvement? Because I was also wondering, are we also looking at a different drug that would just increase the neural connections, which would then hopefully help them get gains?
Yeah, I don't know, because like when we're treating the humans, we're not looking at the brains. All that they can really conclude is how they're doing behavior wise, and their symptoms and everything. So I don't have a good answer to your question.
Yeah, the brain is a really magic organ. You may have heard that people that are blind, have really good hearing, or people that are deaf have really good vision. And so your brain can kind of rewire itself. So, if people are missing a structure in the brain, which does sometimes happen with DDX3X, you may not know what's going to happen if you introduce some kind of gene therapy, because you don't know exactly how that particular brain has been rewired in its own way. So, I think this is why we do the careful research to try to show that things are safe, because that's the number one goal, right? Is to not make things worse. And then what's going to happen in terms of outcomes is going to be dependent on exactly the people and how they respond to it.
Yeah, and I yeah, we can try to explain the mechanism based on whatever we can do in mice, but we're not going to biopsy the brain, to try to look at a cellular molecular level, right? Yeah. 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 And I want to come back. Beth kind of asked, like, why do I feel optimistic? And, one other maybe last comment that I'll make is I think you saw like my lab's working on a lot of different things. We're really, really busy. And I'm really careful to choose what we use our limited bandwidth to work on. And if I didn't think there was a path, we wouldn't be working on DDX3X.
With that, I think we're going to gather up and say thank you to Dr. Gray for a great presentation. And I'll pass it off to Beth.
Thank you so much, Dr. Gray. You're the best. He also needs a lot of money, so he'll tell you what he needs. I said, you need money. Oh, no, he needs. We want to take a picture of all the parents here today. So typically we do it on the steps right before we have lunch. So before you get your kids, if everyone could pause on the steps so we can take a picture of the families here today. We also need a picture of the doctors and scientists. So before you guys start lunch, I'd like you to go more towards the back of the lobby, and we'll get a picture of all doctors and scientists there, all families here. And then if your kid is a DDX3X kid here, we want to go into the babysitting room before they disperse and take a picture of all the kids together, which is always really amusing. Herding cats. So thank you. We're going to break until 1:30, but let's get the pictures out of the way.
I in the meeting? I think somebody else in that way. Yeah, I think the mic works. Do you want me to get started? All right. Hi, guys. I get to bring us back from lunch.
Hi, everyone. We're going to go ahead and get started.
All right. So again, I'm Tamar Caceres. I'm a pediatric neurologist and specializing in neurodevelopmental disabilities, and I am at Rush in Chicago. And so throughout the morning, we've had the opportunity to hear from researchers doing work in DDX3X and discuss opportunities for collaboration. And one of my big focuses right now is the natural history study in DDX3X syndrome. So I wanted to talk to you a little bit about that and what it involves and what we've been collecting over the past 2 years. In addition to a little bit about the differences between clinical care and participation in research. So starting a natural history study in DDX3X syndrome has been a priority of the foundation for a really long time. And with a lot of support from the foundation and financial support, we were able to start that at Rush and start seeing patients in January of 2024. A lot of really great work has been done describing cohorts in description of individuals with DDX3X syndrome in the literature, but our goal is really to create infrastructure to follow people over time in a systematic way, and then hopefully scale that so that not everybody has to come to Chicago or to Philadelphia, since we know that is hard. So we talked this morning a little bit about the role of natural history, being able to collect data over time and understand parent and researcher priorities to better characterize the syndrome and range of severity, and be able to go back and forth that chasm from preclinical to clinical work and translate and back translate and try to collaborate to be able to make progress in the field. Identifying areas of need for therapy and developing and evaluating appropriate outcome measures for future clinical trials. So in any rare genetic and developmental disorder, this is a really foundational part of moving things forward. So as we heard this morning, we know of 1,300 individuals with DDX3X syndrome, and they're all, about 200 of them are described with varying degrees of detail in the literature. And so now we're tasked with trying to take what we know and collect information in a systematic, longitudinal way. So many of the topics that have come up, we know individuals with DDX3X syndrome are affected in a wide variety of ways. And so we are aiming to characterize cognition, language, motor performance, adaptive skills. This is different for every individual. We are exploring symptoms of autism spectrum disorder, behavior traits, sleep came up, and trying to further explore and characterize the genotype-phenotype correlations that have been described in the literature, in addition to looking at quality of life for families over time. So the term centers of excellence is used differently across different consortia and with different foundations. So at Rush, we are a site where people can come and see us clinically if allowed through their insurance, and they can also participate in research as part of a protocol with a consent, and they can do one and or the other. Other sites have varying capacities to do that as well. And so CHOP UPenn is opening as well as a site for the natural history study for individuals as well. And previously UCSF was seeing patients for the natural history study. So when somebody sees me in clinic, that is a clinical evaluation of their neurodevelopmental history, and I provide specific recommendations relevant to them. I follow people over time and provide medical management and can do one-time consultations and annual visits. And so those are part of clinical care. And then I was saying we also have research visits where a specific protocol is followed, and that is per a consent process. So participation in research is always voluntary, and for the natural history study in DDX3X syndrome, it is agreeing to share de-identified data for future research. We are really working to have what's called globally unique identifiers, like de-identified identifiers that could link data from different projects to each other, because we know that once somebody contributes their data and we go through the process and burden of doing that, we want to make sure that that data will be accessible and can translate to other researchers, or how it might be as we go forward in the future, how that might be relevant. So like we talked about, these are some of the clinical things that we do in evaluation of somebody with DDX3X syndrome. And And then we have another process for participation in research and enrolling in our natural history study. And in Chicago, we coordinate these, right? So you don't have to come see me once for a clinic visit and then another time for a research visit. We do them on the same day as part of the same process. It is just participating in two different types of processes. So this is a link to Melitza, who's our research coordinator, who does all of the coordination for all of our clinic visits and our natural history study at Rush in Chicago. She's sitting next to Juanita. Who is a geneticist here from CHOP at the UPenn site. And so we also have contact information for their research coordinator if you think that going to Philadelphia might be more convenient for you, and we can help with that process. So for participation in the natural history study, you have to have a diagnosis of DDX3X syndrome. If you aren't sure if you have a diagnosis of DDX3X syndrome, you can send us the genetic report and we can help you figure out if you are in that category. And right now, we do not yet have the ability to be providing financial support. So we recognize that is a burden, and have been really impressed with the community's eagerness to participate and families coming to Chicago and coordinating that travel and cost. So the development of the protocol for the natural history study, what assessments we're doing and what information we're collecting was developed and informed by what we know or knew about DDX3X syndrome through existing literature and prior projects and clinical care expert experience. And so a few years ago, we were in the process of all meeting and discussing what tools we were going to use as part of the natural history study, and we're still learning. And so, sometimes we are needing to update because the tools that we were attempting to use didn't actually make sense in the population once we tried collecting them in a systematic way. And so we've needed to make modifications as we go through this early learning process. So this is an unpublished caregiver report symptom survey that was completed by the foundation in 2020, looking at concerns and symptom presentation. And so we took this information and wanted to make sure that we were capturing these things in addition to things that had been published by clinicians and researchers in the literature. So currently what is involved in participation in the natural history study, it involves traveling to the site that is collecting the data. So currently that is Rush in Chicago or CHOP UPenn in Philadelphia will be open soon, hopefully, to start enrolling patients. We are collecting new data over time. So individuals have been involved in lots of amazing projects previously. When you participated in those projects, you didn't necessarily consent to share that data with the natural history study. So we don't yet have an ability to pull that data over, and this is new information collecting going forward. At Rush, typically we are doing in-person 2-day assessments. For some families that doesn't make sense and we need to spread it over 3 days. And so we have some flexibility in how we coordinate this. All information, whether it was previously collected at UCSF for this natural history study, or it's being collected at CHOP or at Rush, we are all entering the information into a centralized database where, again, things are de-identified and linked to a unique identifier for that participant, with the goal of developing a system where that will also at some point feed back into the foundation registry. So as part of research, as we were talking about earlier, we go through a detailed consent document and everything's explained in detail of what's planned for that study and how we collect the data. And there's a system that we use through these unique identifiers to link research data and specimens in databases across sites. And processes of collecting samples when indicated to explore future biomarkers of disease. So currently at Rush in Chicago, we aren't collecting blood samples from our patients with DDX3X syndrome, but other sites have in the past and will in the future. We're running a EEG study today. This is actually a parallel study to the natural history study, but we may build that in and just do it jointly as we go forward into the future. So what are we collecting? We're doing detailed assessments ranging from medical history, a specific exam, parent checklists, again, looking at symptoms that have already been described. So a lot of what we're seeing early on in our data of what we're collecting is confirming what is already described. So it's not necessarily new yet. We're just trying to collect it in a coordinated manner so that we can follow people over time. We are administering parent surveys about specific types of symptoms through an electronic system that all feeds directly into the confidential database, and then are doing performance-based assessments where a trained rater is working with the individual with DDX3X syndrome and doing evaluations of motor skills, language skills, evaluating features of autism spectrum disorder, evaluating fine motor skills, and also a clinical evaluation with a pediatric neurologist or somebody experienced in care for individuals with DDX3X syndrome. So we did this for the initial year in 2024 and found that not all of the tools that we were using were working. And so we needed to have some modifications to the measures we were using. And we want to better understand not only how individuals perform on these measures one time, but how they perform on these measures over time. So these are all performance-based assessments. They are assessments that are done specifically with the individual with DDX3X syndrome. And it sort of provides an overview of the time that it takes to do those. We take frequent breaks. We have a lot of experience doing these types of assessments in individuals with developmental disabilities. And some of the measures are more or less challenging. We have a whole process of explaining this to families. Based on somebody's abilities, they would do an assessment from each category. So you do a cognitive assessment. Everybody does what we call the NIH toolbox, which is a measure that's been developed to look at cognitive abilities and problem solving specifically in individuals with developmental disability. They do a number of different language assessments, which I'm not going to present a whole lot of the language data here yet today because it's so complex that we have a lot more to need to look at before we're able to present it. Motor skills, and a psychologist evaluation of autism spectrum disorder. And these are all the different types of parent forums or clinician forums that we are looking at, including adaptive skills, communication evaluation, social skills, challenging behaviors, sensory features, sleep, and quality of life, again, to see how what results from these measures look like in this population and seeing if any of them would potentially be amenable to be used in a clinical trial. In addition, as we mentioned, sometimes we do additional testing as well, which is outlined in either the consent for the natural history study or if you consent to participate in additional work. So for example, blood draws or EEG. We talked about how we store that data so that it is available in its de-identified form to be shared with future qualified investigators is the goal. And following people over time and then being able to explore specific subgroups or specific age populations as we go forward. So at this time, right, this is really an investment in the future of research in DDX3X syndrome. We're able to provide a basic research report of scores from the assessments, but they don't include clinical impression. And so, we really appreciate your participation. So at Rush, we have seen 26 individuals since January 2024. Between 0 and 25 years, we have seen 24 women and 2 boys. We have seen 1 person for their follow-up assessment, and this is a breakdown of ages. We've seen people across different genotypes, but notably we haven't seen people with all of the variants that we are looking at in mice as part of this natural history study. And so that will be an area where hopefully we are able to learn a lot of good information. And this expected impact, we talked a lot this morning about the underlying mechanisms. And so this is not functional analysis of how that variant actually impacts that person, but it's what we would expect to happen based on what their genetic change is. We are seeing a number of recurrent variants so far in our small cohort. Again, these are not all the recurrent variants that are reported in the literature, just the ones that we have enrolled so far in the natural history study. And I'm going to go through some initial both clinical and developmental data. Again, this is a really small cohort of people. So we have fewer numbers than what already have been published in the literature, but it helps us get an idea of what we're seeing and if it's representative of what's described so far in DDX3X syndrome. So this includes people who were seen at UCSF for the natural history study, in addition to people who were seen at Rush for the natural history study. So far, all individuals have been born term or late preterm, and about a third of them were in the NICU as part of their care early on. I have picked what information to pull out and present. So if there are specific parents or clinicians or researchers who are interested in specific information, you can always reach out to me and say, hey, can you pull this out? And, let me know what it shows or present it at a future meeting, because it's something that, really interests me or something that really affects my child. And are you collecting this information, or do you have this information? And sometimes, if it's something that we're not collecting and it's meaningful for the community, we have the ability to modify as we go forward through a consent process and an update process of how the protocol or measures or questions have changed since you enrolled in participation. So looking at caregiver reported early infancy medical problems. Again, this is not all, but there is a high rate of reported GI issues in the newborn period, about 20% reporting respiratory issues. And again, this is when a clinician is interviewing the parent and asking specific details about types of GI or respiratory symptoms. Did your child have reflux? Did your child have constipation? Did your child have pauses in their breathing? Did they have cyanosis? And then we are recording that on a standardized form and entering it into the database. And this is currently out of 30 individuals. The percentages look pretty similar to when it was out of 20 individuals when I presented this last year. And so it helps us get an idea of if we think that it is a representative population. Going on through early infancy, starting to look at some additional behaviors. We are seeing most commonly early things that are described are hypotonia, poor feeding, with about 70% of people having low tone and 50% of individuals having difficulty with feeding, in addition to symptoms like reflux or decreased movement or decreased alertness. When we start to look at gross motor milestones, we're now seeing people of all different ages, right? So when I see an individual who is 6 months old, they are not pulling to stand and not walking yet. And so depending on how we're presenting the data, that can be relevant. So they have not yet been expected to meet that milestone based on what we would expect with typical childhood development. So for this cohort, individuals who were less than 24 months old were not counted in these percentages based on what we know in the condition so far. So we have a wide range of acquisition of developmental gross motor milestones with individuals between 3 and 20 months of age achieving rolling, sitting between 8 and 24 months of age, again, crawling between 12 and 30 months of age. Crawling is not a required expected developmental milestone. Not all people crawl. It requires a lot of coordination. But we included it there, pulling to stand between about 1 year and 3 years. The majority of the individuals enrolled in the natural history study so far walk, with about 70% achieving that milestone by 36 months of age, about 3 years of age. Certainly some individuals achieved that milestone earlier, but 70% are able to walk by that age, and almost 90% who are able to walk by almost 4 years of age. But again, the 50% walk between 15 months and 2 years of age so far based on what we're seeing. Toilet training has big impact in quality of life in individuals. So, this is something that we are collecting specific information on. These categories aren't necessarily individually exclusive. It's a check all that apply question. So you may have somebody who is fully continent during the day, but they need to toilet on a schedule. So we have a third reporting that they are able to toilet train with some nighttime wetting, a third that are fully toilet trained, and a third that are not toilet trained at all. This is of 21 individuals who are over the age of 5. So again, based on what we know in the condition so far, using that age to comment on how many individuals, what is the toileting profile of individuals who are in a early childhood group. We know that language is significantly impacted in DDX3X syndrome, and we are still looking to characterize what the contributing factors to that are. So this is looking at 25 individuals in the Rush cohort. When we describe language skills, the standard in the literature is to define what we call minimally verbal as having 50 words for a child who's over 30 months of age, or single words and simple phrases. So saying somebody is minimally verbal does not mean that they do not have any words or do not use speech to communicate. And I don't have this cohort separated by age, but there's about 64% with either no words or just single words or simple phrases, and about 36% with longer phrases or sentence speech. And that's been described previously, that there are about 50% of individuals with DDX3X syndrome who speak verbally and about 50% who don't speak verbally. And we are now trying to get a little more detailed in our understanding of that. I'm a neurodevelopmental neurologist by training. And so I want to know, like, what does that mean that people speak sentences, right? Like, are they speaking sentences at the level of a 3-year-old? Are they speaking sentences at the level of a 16-year-old? And what does that mean functionally in terms of communication, right? You can get a lot of information across with 3 to 4 word phrases, even if you don't have some of the nuances of more advanced comprehensive language. We also know when we ask people which of these are a primary modality of communication, 55% of a group of 22 will say that speech is a primary modality. Gestures and sign are a primary modality in 73%. And augmentative communication is a primary modality in 55% of the population. And so we can use this information to guide clinicians, right? We don't necessarily have to wait for individuals to not be effectively communicating verbally at 3, 4, 5 years of age. We can introduce augmentative communication strategies early on because we know that this is a population that is vulnerable in terms of their communication abilities. And there's good evidence that using all strategies for communication continue to support the development of speech. So introducing sign language or a communication device does not mean that you are hindering your child's speech communication development. Looking at prior caregiver-reported diagnoses, similar to what has been characterized previously in DDX3X syndrome, we are seeing that almost all individuals somebody has previously told them that they have global developmental delay. Again, it depends on age, right? Because our clinical system and the process of being evaluated, you have to miss milestones before you receive a diagnosis of global developmental delay, and then you have to be delayed in more than 2 developmental categories. About 60% have diagnoses of intellectual disability. Again, we're seeing patients across the age span. So somebody that we see at 1 year of age is unlikely to have been given a diagnosis of intellectual disability yet, although because they have a genetic diagnosis that is associated with intellectual disability, depending on who they were evaluated with, that may have been discussed or not. And so we can use this also to make improvements in the clinical care that we are able to provide. About 70% of individuals are reporting movement disorders. This includes things like spasticity, tics, ataxia, apraxia, and we have the specifics of all of those different sort of subsymptoms. But it would take me forever to present all of the data. So we've grouped things into categories. We heard a little earlier that sometimes people get a genetic diagnosis and then they don't necessarily have symptom characterization or diagnoses given related to the symptoms they're having. So for example, epilepsy is a symptom, right? That means that you have seizures. They are probably due to something. Autism spectrum disorder is a symptom. And so you can still have autism spectrum disorder or intellectual disability or epilepsy or ADHD due to DDX3X syndrome. Notably, we haven't seen anybody beyond the age of early 20s. And so we haven't gotten to do a lot of characterization of mental health concerns that may come up, as is more commonly reported in adults with intellectual disability. And so we don't yet have anybody where parents have been told or caregivers have been told that there are issues with depression or OCD or schizophrenia or things like that. But again, so far, people enrolled have been in the younger groups. When we look at mood and behavior symptoms, again, this is just based on what symptoms parents are reporting. We see high rates of attention problems, differences in sensory processing, agitation, and hyperactivity and sleep problems come up again. So to try to better characterize some of these caregiver reported symptoms, we are using parent scales that are validated in populations of individuals with intellectual disability, and there is literature published on in related conditions or in grouped conditions like individuals with autism spectrum disorder. So we are collecting a parent survey about what the sleep behaviors are. This is a 45-item questionnaire where parents are reporting how often their individual with DDX3X syndrome experiences these symptoms. And traditionally, a cutoff score of 41 was used to indicate when there is a high risk that based on that report measure, those individuals have sleep problems. The measure was explored in individuals with autism spectrum disorder by age, and it was found that using a lower cutoff would likely be more appropriate in that population. So based on age, using cutoffs of like 33 to 35 based on age would better capture sleep problems in that population. So when we look at the measure so far in DDX3X syndrome, again, this is only 19 individuals, but we're seeing that their range of scores is between 32 and 64. So almost all of them are where we would start to be concerned that there is a sleep problem, and that they had an average of 44, which is above the cutoff that was used initially in the development of this questionnaire and the validation. We also are doing a specific parent questionnaire trying to explore sensory processing, since we know that there is high pain tolerance, there's hypersensitivity, and I don't know if there's any OTs in the room, but sensory information is really complex. How this measure is designed is it classifies individuals into quadrants of style of sensory pattern. So there are individuals who require more input to react to a stimuli, who then may seek additional input to have the same level of sensory processing. And so those individuals might be classified as seekers. Avoiders may be sensitive to extra sensory input. Bystanders may not notice certain types of sensory information. And again, this is really complex. I'm working with an OT, and often we look at whether there are overall patterns. Are you likely to be are you likely to fit into multiple quadrants for their sensory data? So this is just looking at the quadrants individually, and we have about half in each of the categories that are having these types of sensory patterns. When I'm saying about half, we are describing how many of the individuals are greater than 1 or 2 standard deviations from the mean. So are in this category of being more or much more than others to have that symptom. Looking at the other side of things, if you are less or much less than others, will also be interesting in exploration of this sensory information. When we look at medical problems by system, we are seeing high rates of GI symptoms. It's very common to have vision symptoms. I'm not able to see my breakdown on the slide because I can't see my notes for whatever reason. But we are able to look at how many individuals have cyclic vomiting, how many individuals have constipation. A high rate of individuals wear glasses. And we are able to take this information to supplement what has already been put together as part of practice guidelines for clinicians taking care of individuals with DDX3X syndrome, right? So everybody should have early screening. Everybody should have hearing tests, not necessarily just once, but potentially over time. There is a high rate of recurrent ear infections. A large number in our cohort have had ear tubes. We know that there is a association with congenital heart defects that often are simple, but sometimes are more complex. And so how do we integrate this into standardized clinical practice to be able to provide individuals with the most appropriate care? As we know, my lens is from pediatric neurology, and so currently in our cohort we have about 37% who have a history of seizures and 70% with movement disorders, and again that includes a wide variety of things that fall into the category of movement disorders. So we've started to look at some of our performance-based data and some of our other interview collected data. So adaptive functioning is it's standard to evaluate with a measure called the Vineland, which is a caregiver interview of on average how an individual performs in certain aspects of their life in terms of taking care of themselves. And so far, we see a wide range of functioning across all domains of adaptive functioning, but generally within the range of intellectual disability. So around 100 is considered to be age appropriate, and things below 100 are considered to be delayed for age. And this is a measure that is not able to capture below a score of 20. And so we see that many individuals are what we describe as at the floor of this measure. So we aren't fully capturing their abilities with this measure. We see, if you look, we have these little lines in the middle. So these are averages. And in general, socialization is a relative strength. And communication is a relative weakness in comparison to overall adaptive functioning. We have these dots color-coded by type of genetic variant, and we are seeing different colored dots throughout the distribution. As I mentioned earlier, we have not enrolled individuals with all of the different types of variants, and we haven't enrolled individuals with all of the different types of variants that we have mouse models of and that have previously been implicated as having a certain degree of severity. And so it will be interesting to see if there is a shift in this data once we are able to enroll individuals with those other variants. When we look at how individuals function in the real world, in comparison to their IQ scores, we see that their day-to-day functioning is a little better than we would expect based on IQ scores, right? So IQ scores clinically are not so relevant, right? We want to know how are they doing with their daily activities and how can we help support them in their daily activities. So we see that their adaptive scores are a little better than their IQ scores on average. Again, we see a distribution across different types of genotypes. And we see, as we would expect in a population where expressive language communication is challenging, we see a little bit of a split between nonverbal IQ and verbal IQ, with verbal reasoning being a little less well developed than nonverbal reasoning. And the distribution of functioning being a little more distributed toward the lower end of functioning with regards to verbal IQ. We are using different measures to assess individuals of different abilities based on the tools that we have that can assess problem solving and cognitive skills individuals with developmental disabilities. So these were developed in typically developing individuals, and there has been a lot of work in the field to figure out how can we most appropriately assess individuals with developmental disabilities, what tools can we use to better understand their abilities and making sure that we are best capturing those. So we then take those scales and do a process so that we can collapse them and look at them together. One of the areas that I have recently been exploring is looking at how features of autism spectrum disorder present in DDX3X syndrome. And so some of this we presented recently at the Child Neurology Conference. We know that individuals with intellectual disability may have some symptoms of autism spectrum disorder related to their overall cognitive ability. But what does that mean? And we also know that individuals with intellectual disability can also benefit from autism-targeted therapies, even if they don't have diagnoses of autism spectrum disorder. And so what does that mean for the DDX3X population? So we were looking at the social communication questionnaire, which is being collected as part of the natural history study. This is a parent measure looking for symptoms that identify risk for autism spectrum disorder. The ADOS, which is a psychology administered play-based tool looking at how an individual interacts and plays with a psychologist who is typically somebody they have never met, and it's done in a room where they are unfamiliar, and how that compares to the gold standard, which is a clinician diagnosis, taking in all of this information in addition to what parents describe, who has training and expertise in the evaluation of diagnosis of autism spectrum disorder. And so we find that scores above 11 on the SCQ raise concern for autism spectrum disorder. So when we look at how the SCQ is categorizing people in comparison to how the ADOS is categorizing people, we see general alignment, that the ADOS is identifying that the individual likely has autism or autism spectrum disorder in the individuals who have scores above 11 on the SCQ, and in general, individuals where the ADOS scores them as likely not being on the autism spectrum disorder generally have lower scores on the SCQ. We found that there is a high rate of false positives in both the ADOS and the SCQ, where individuals who were found to have autism spectrum disorder based on those measures are not found to have autism spectrum disorder when evaluated by a clinician who has experience in the evaluation of autism spectrum disorder. And so why does this all matter, right? It depends. Sometimes a diagnosis of autism spectrum disorder is needed for access to services. Sometimes people get a genetic diagnosis and then don't have evaluation for symptoms of autism spectrum disorder. And so we're trying to understand how these different measures perform in this population. So when we look at features of autism as scored on the social communication questionnaire, in addition to cognitive functioning and adaptive functioning, we see that there is a greater correlation, a stronger correlation between adaptive functioning, how you perform in your everyday environment, and features of autism spectrum disorder. There is a correlation between IQ, but it's not as strong of a correlation. And so that's suggesting that individuals with features of autism spectrum disorder, even if they don't clinically have autism spectrum disorder, that is potentially having a greater functional impairment on their everyday lives. And again, these are small groups of 22 individuals, but it suggests that those features of autism may have greater functional impairment. We also looked at this in terms of other measures of behavior using the aberrant behavior checklist, and we're able to see that there is a correlation also in terms of how those other behaviors impact adaptive functioning, but those correlations weren't as strong as they were with with features of autism spectrum disorder as measured by the social communication questionnaire. We've started to look at quality of life. Again, these are all measures that are collected by the Natural History Study. So this is a tool that was developed looking at parent-reported individual quality of life. Higher scores are suggestive of higher quality of life as reported by parents. We see that, you know, there are sort of high highs and low lows, but social interaction and total score still are distributed toward higher areas of higher quality of life. And we're also looking at a family quality of life measure and seeing how this looks like in different domains of quality of life. Quality of life is really important to, you know, to understand when we're thinking about improving outcomes in individuals. So related to some of the things that we were looking at before, where we were looking at individuals with greater features of autism spectrum disorder having potentially more functional impairment in their day-to-day life, we also see a strong correlation between quality of life and symptoms of autism spectrum disorder. So that's a little overview of some of the information that we've been able to pull and look at so far from the 30 individuals who have enrolled in the natural history study. My list of collaborators is growing, which is really exciting. I have my whole team here from Rush today and have lots of students who are involved in starting to pull apart and look at some of this information that we're collecting. And we are so appreciative of all of the families who have schlepped to see us in Chicago, and all of the individuals who are going through, you know, sometimes non-preferred activities. We try to make it fun and preferred, and they've really, you know, done amazing. And then, of course, thank you to the foundation who is funding this project. So I don't know if now do we have time to open it up for sort of general clinical questions, or what's next?
Bring the mic up.
Thank you, Chelsea. Thank you. Nice job. Really appreciate it. I have a question that it might be personal, but I think it's from some of the OGs that might be able to help answer it. And I'm putting my dad of the year at risk here by asking this, but I'm not sure if we're in the study. The original one is in Pittsburgh. Was that a study?
Yeah, so there was an initial excellent characterization of an early cohort of individuals. They were not able to continue to follow them over time. So those individuals, unfortunately, are not captured in this information. We would love to have you involved.
Do I get a free trip to Chicago because of that, or?
If you can chat with the foundation and you can have a cook-off. But there are individuals who have enrolled at UCSF, and so those individuals are in this study, even though UCSF is not able to continue to be a site. We are trying to, for the future, be able to link data together. So hopefully, if you know of researchers who are studying individuals with DDX3X syndrome, we want to use the model of using globally unique identifiers. So individuals are assigned a de-identified identifier that then could link them across projects. So if you contribute data, a researcher looking at something else would be able to collect new data and look at data that was previously collected. As we've talked about, there's lots of consent processes and regulatory regulatory measures to protect patient information, and we want to make sure that you understand what is being done with the data, how it's being stored, and who has access to use it in the future, because that is all your right as participants in research.
Long time getting the list of tests that all the sites would agree to across the board so we could have standardized data that made sense, that was consistent with the same battery of tests. And so what we did at Pittsburgh back in the day wasn't the same test necessarily, some but not all of them. And unfortunately, she stopped studying it before we could get it into this FDA approved registry.
Got it. Thank you.
But if anyone wanted to go back to Pittsburgh and see if they want to reconsent to share their data.
Quick question about the vision. It seemed like a high percentage of people have vision problems. And with our daughter, who's mostly nonverbal, we've never had success testing her. I noticed some young ladies are wearing glasses. What is there one specific vision problem associated with DDX3X? And is there a method, if we don't live in Chicago, to have her properly tested?
So far we are seeing high rates of strabismus where the eyes do not align. And then also high rates of hyperopia or myopia, like where you have refractory error, you need glasses. So ophthalmologists do have an incredible ability to assess individuals who are not verbal and who can't participate in testing. There are 1 and 2 month olds who wear glasses or have been identified to need glasses. And so you don't have to come to Chicago, but you might need an ophthalmologist who has experience working with children to do that evaluation. We aren't directly doing vision testing. All of our information about eyes and vision is parent reported.
I'm sorry if you've already said this and I missed it, but so my daughter turned 2 in July. Is someone like her, like, is that too young? Like, at what age do you recommend starting? And then with her being so young, like, how often are you wanting to have these patients come back and do that as well?
Yeah. So we would love to evaluate anybody of any age. Some of the measures are not yet appropriate based on age, although the majority of them are. And so based on the age of the participant, they wouldn't do the measures that aren't yet appropriate for their age. And as they continue to come back, then they would start doing those measures. Because of what we anticipate with regards to typical development, which we don't have the developmental trajectory yet understood for DDX3X syndrome, but we are aiming to evaluate the younger individuals more frequently. So we are aiming to evaluate individuals under 6 every year to try to better characterize those early developmental stages, and individuals over 6 every other year. But you're welcome to touch base with Melitza. We also, understand that this is a burdensome process, right? And so if you missed a year, that doesn't mean you can't come back, right? Like we are working with people to try to make this feasible, and hope that opening up at CHOP will be able to target some different coasts, different parts of the United States.
And then one more question, if I may. It seems like, I think it was one of the first graphs you showed of like all the different symptoms. For someone, again, this might be more of a personal question, but for someone like me with such a young daughter who currently right now, for example, vision being one of those things that's there, she doesn't exhibit issues right now, but would you use that or recommend that from a clinical perspective to maybe like take that a bit more seriously or look into it a little bit sooner, because that's potentially a path that she might take? Same thing with like seizures and that sort of thing.
Yeah, so information that we have so far about DDX3X syndrome, they've been able to put together some recommendations for clinicians with gene reviews and that recommend evaluations of vision, evaluations of hearing, evaluations of baseline cardiac evaluations. And so even if you are followed by a provider who doesn't necessarily have a lot of experience in DDX3X syndrome, those resources are available, and the foundation or other people can help connect you with them. It's challenging, right? You all have very busy lives and individuals with special needs in your family. And so we understand that there is a balance of trying to juggle, what do I need to do now? But if you are able, those are the things that we've identified as areas that might have targetable interventions, right? I recently saw a young individual who the family described, and again, I'm just using this anecdotally, this doesn't mean that it will happen for every person, but the family described that as soon as they got glasses, they just all of a sudden were interacting socially in a way that they had not been before. And so it may not always be a vision problem, but sometimes it might be. And similarly for hearing, right, we want to make sure that there isn't a tool that we can use to support your communication and language development. And so if there is a possible contributory hearing problem, like that's something that we want to identify early and intervene, even if you are have an underlying genetic disorder that is increasing your risk of communication challenges.
Thank you so much for your talk and all of your research and efforts for all of us. It's so validating to hear things like this. We're still in the process of having gotten our diagnosis only a few months ago, asking our GP for ENT referrals and being declined, that kind of stuff. So this is super validating. My specific question, and I think I'm sure a lot of us can collaborate here and say that I've gotten so much more of my information from the Facebook group than I have from my pediatrician. In terms of something that I don't believe that you touched on, and I'm curious if you've seen any patterns, is teeth. Something that's been unusual is our 18-month-old, her front teeth are all decaying and falling apart. And it's not something that's come up in the literature. Would love to hear what you found.
Yeah. So this is something that we are collecting in our sort of clinician interview, and in addition in our physical exams. Because we heard of individuals having experience with this. So I don't have specific numbers for you. It's definitely something that we can pull out. But we have heard that there are potentially issues with enamel or issues with crowding. They're also structurally, for some individuals, they have small jaws, they have differences in their palate development. And so that certainly also has implications for dental, and teeth development. And so early dental care, if we're able to, if any of you know a dentist who has interest, right, like that we have a lot of room to improve dental care in individuals with intellectual disability in general, and then in conditions that it's been identified to have potentially unique features.
I'm glad that you said we should find a dentist, because I don't know if anyone else saw on Facebook recently, there was another parent in Canada that just got a diagnosis who's a dentist.
Exactly. But
So
pull
I'll message her.
your areas, right? Because if you have a specific connection, then you may also be able to make change in your area.
Thank you. I'm happy to bring the mic to anyone else.
So much. We're going to do the parent panel now. So we're going to gather the parents who are speaking on the panel to come to the front. If you guys want to take a quick break, that's okay. We're going to start in a sec.
Hello. Oh, yeah, great idea. Okay. Hello. Okay, great. Hi, everyone. This is our second annual parent panel. We want this to be really participatory. This is a chance for all of us parents to ask each other questions, and some of us OGs can pass on the information that we've learned over the years. Beth unfortunately had to leave, but she will be back later tonight. So I just wanted us to first start with introducing ourselves and our child's age, name, and presentation. So my daughter is Lily. She is 12. She is very cute, nonverbal, fine and gross motor challenges, no real behaviors. But I would definitely say the communication is our biggest challenge and hurdle to deal with. First dad.
Hi, everybody. I'm Nick Tambone. My daughter is Lena. She is 11 years old. Nonverbal. She does walk, but she has some motor challenges. Communication is probably our biggest issue. We are seeing as she gets older, definitely more opportunities when it comes to anxiety in different situations. Unfortunately, often culminating in cyclical vomiting, which if anybody's dealt with that, lots of fun. So, that's been a big thing. Overall, though, mainly healthy, no major health concerns or risks there. But, like I said, definitely communication's our biggest thing. And then I was told I had to mention Kate, my wife in the back there, is on the leadership council. She was talking with Ursula, who is a DDX3X mom from Poland, who went back to school and got her doctorate after getting the diagnosis, is running her second survey right now. So it's for kiddos with DDX3X or autism, or both. So if you would like to take part in that, you can either see Kate afterwards or jump on the Facebook site and there's a link to it.
My name is Lindsay Grace. Emma is my daughter. She's 10 years old. She's extremely verbal. She pretty much doesn't stop talking, which is great most of the time. A lot of time I'd say her anxiety appears and she will ask the repetitive questions just over and over and over. If you turn around and ask her, she 99% of the time knows the answer, and then she'll ask it again in about 30 seconds. I attribute that to mainly anxiety kind of stuff. I'd say fine motor skills are still very difficult for her. Any writing, things like that, she doesn't like to do or even practice. She does, but she would avoid it. Maybe 3 or 4 months ago, I would have told you she was doing really well, walking great. We've been in physical therapy since she was 10 months old, but she's come a long ways physically. She's developed a limp and has fluid and inflammation on her hip. We've seen everyone and done everything, and they don't know why. And so she does have a limp now, and we're using a medical stroller for anything, long-term walks or anything like that. So we don't know if that's connected or not, or positionally, because again, we've done all the things and haven't found an answer yet. So beyond that, generally normal kind of health sicknesses and things. She does deal with chronic constipation, so that's something that we have to stay on top of all the time. But, oh, she's had the strabismus, she's had surgery for the eye, but I'd say everything else we've dealt with, like ear infections and things like that, have been pretty normal.
Hi, I'm Jamie Sneed. My daughter is Emmeline or Emmy, and she is 12. She's had her diagnosis for over 10 years now. And I would say healthy for the most part. We also deal with constipation. And we our new battle right now is spinal and skeletal issues. She's got a nice S curve in her back, and so she sits at kind of this 40-degree angle, and so that's our new thing is how is that going to develop as she enters puberty. Obviously, puberty is also one of those things that we are on the cusp of at this point, and how that's going to affect her. She's a happy kid, has never met a stranger, 100% nonverbal, but has 500 plus signs. Many of them are animal related signs. That's her happy place. She reads probably at about a first or second grade level, and loves words and books. And so that's been a great thing for us to see her intellect come out and her ability to read. That being said, talking about our kids being kind of scattered, she has refused to learn money up until recently. So math, not so much her thing, but she is bright and is teaching us lots of really wonderful things.
Hi, I'm Melissa. I'm Annabelle's mom. Annabelle is 20 as of Sunday, which is very strange for us. We were diagnosed in 2015. Annabelle's nonverbal. She communicates with a talker, an AAC device on an iPad. She has a lot of anxiety, OCD, maladaptive behaviors, but she loves animals. She can name so many animals on her talker. She loves to talk about animals, especially animals in Go Diego Go or Dora or any other cartoon that you can think of. She knows every episode of Phineas and Ferb. But yeah, it's been a long journey.
Hi, I'm Sarah. My daughter is Olive. She is 10 and a half years old. She has bright red hair. She is verbal. She is, like many of these other children, very happy, very sweet, very smart. She taught herself to read at 4. She has an amazing memory. I think her biggest struggles are probably daily living skills, fine motor. She definitely has anxiety. Thankfully, she went on anti-anxiety medicine, and I'd say it's helped at like 75%. So that's one battle. And, overall, she definitely struggles with day-to-day living, but she's improving. So she's on the right trajectory.
Thank you. So because there's a lot of new parents here, I thought it'd be helpful if we talked about lessons learned that we've learned along the way, or advice that we would want to pass on. So 2 things for us. One, we hear a lot about the GI issues that she was constipated, she had diarrhea. It never occurred to us that she had a dairy allergy. So that's something that I wish we looked at sooner. So I would recommend that being put on your to-do list. The other thing that we didn't prioritize was cortical vision impairment, which we're now kind of diving into right now because she had so many therapies and so much to deal with. We're like, she can see fine. But that's actually not about vision. It's how you visually process the information. So we're sort of on this journey now that is impacting which AAC device is best for her. So we're figuring that out. So just 2 pieces of wisdom I'd want to pass on to everybody.
Wow. So many different things. One of the biggest things I'd find is kind of constantly challenging what they can do. So I just find that I've fallen into at times kind of accepting, okay, Lena can do this. She can't really do that. than getting kind of hit over the head with it when we talk to her teacher. Oh no, she does that at school. Nope, never seen her do that. You know, she has an AAC device. She has no interest in using it. Very basic, you know, one of two choices. Apparently she does it at school. Won't even look at it at home. I give it to her and she puts it down. So now we start pushing that more, and all of a sudden she's actually answering questions. And she even referred back to something. Kate went in and read a story to the class. And the other day, our respite worker asked her about it, and she said, "I like," because she remembered that her mom came in and read her a book about Frida Kahlo to her class. So it's one of those things. I didn't think that was something she could do. I didn't think that was something we were pushing towards right now, because it's kind of, you fall into that. I hope the best for her, but I also kind of accept certain things. Don't stop challenging. You know, I'm sure we'll talk a lot about different medical things, different specialists to see all of them. Do everything you can. Everybody who brings it up, try it. But I've just been amazed that never settle, because these kids keep working so hard. Like, I've never been as impressed as watching Lena. She didn't walk until she was almost 6 years old, but we always knew it was coming. Right? I mean, how many conferences did we come to with varying levels of walker for her? When she's strapped in, and then when she's got just a little, you know, rift in one that she's running around when, you know, the other kids weren't stealing it from her because everybody wanted to try it out. But just keep encouraging. I know it gets frustrating, but I'll tell you, when you hit those milestones, even if they're years late, so much more exciting. So many more people are cheering you on. And just these kids, just, I don't know, she keeps amazing me. So.
Yeah, I definitely agree with that. Just recently, I got involved with something called Partners in Policy in Oklahoma, and it's specifically teaching parents or those involved in the disability community to be advocates and how to relate to policymakers in your state. We actually had a speaker come from Pennsylvania and speak to us, and he said everyone's goal should be independent living. And I'm kind of like, oh, I don't, you know, as a parent, you're like, well, I'm kind of preparing myself that that's not going to be the case. And just kind of along the same things. He said you should prepare yourself that your kid can be an independent liver. And I still kind of go back and forth, you know, because I want to be realistic with myself too. But along the same lines is, you don't know what your child will be able to do or not do at this point. If it takes them longer, it takes them longer, but assume they will get to a point where they can communicate. Maybe not verbally, but they will be great communicators, and they have so much in their head. And I know having a young child and hearing all the negative things, sometimes those stick with you more. But just remember that whatever state they're at now, they will continue to learn. They will continue to grow. And you don't know where they're going to end up. So just be prepared that they will change and grow. Something somebody else asked earlier that I had a thought on was taking them to kind of like the list of things that, you know, can happen or that kids deal with. Personally, I wouldn't take them to every single specialist. But it's kind of like if you see a glimpse of constipation or something, don't ignore it the first time you see it, because it probably might be a problem. If you see something with their eyes, that a lot of us that have kids, we're like, oh, okay, well, that was just a one-time deal. If you see it's something with your kids, it probably is not a one-time deal, but I wouldn't take them to a cardiologist if they don't have any issues or haven't seen any issues with the heart or something like that. But don't ignore stuff when you see it. Parents typically know their own kids, and the doctor may not listen to you the first time because they don't know your kid and don't see your kid as often. So sometimes you do have to push a little bit. It's like, no, this isn't normal for my child. This is different. I know my child. And so don't, I guess, be scared to advocate for them, even because most doctors that I've dealt with, I'm explaining to them what our issues are, what the community's issues are. Because if they've seen a DDX3X kid, it's maybe one, maybe two. They're not specialists in that specific area. The parents are.
I'll piggyback on that and say our pediatrician and our former developmental pediatrician before he retired would say the first thing out of their mouth, have you been to a conference lately? What have you learned from other parents? And find that doctor. If your doctor is like, well, I don't care what the parents say, I want to hear what the research says. You want a doctor that's wants both of those bits of information, because sometimes what you learn from the person sitting beside you is not going to come out in the literature for another year or two or longer. So piggyback on that. My two things were, for those of you who have younger kiddos, and I want to reiterate what was said earlier about sign language and AAC devices is starting early is not going to inhibit their ability to gain speech. We started with something really motivational with Emmeline, which was reading. Every time she would hand us a book, we hand over hand signed the word read, 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 until it took 48 hours. That was it, because that was so motivational to her. And from there, it just grew. Her second word was table. So really, that is a powerful tool in giving children language. Emmeline is 12 and still has no words, but has 500 plus signs. And then the second one is Emmeline was 5.5 before she started walking. And I think what gave her and us the final push to get her walking was actually one of those little soft helmets that we could put on her, because we had 2 other very busy children. And so we were always like, oh, you're going to fall, sit down. Oh, you're going to we put that helmet on her, and it gave us all of this peace of mind, and she started walking. So sign language and helmet are my 2 words of wisdom for you.
I love that. I would just echo that. For communication, that helped everything. It helped the anxiety. It took years, but it was worth it every step of the way. And it was constant. It was the parents, it's your team, it's school. They have to be on board. If you walk into your classroom and the kid's iPad is over there, you're in the wrong classroom. So yeah, you just have to keep on that and be consistent, and buy the Apple Care, because it is so worth every penny. Oh yeah, we're regulars at the Apple Store. They're like, "Hi, Annabelle, how are you?"
Something that really helped Olive when she was little was prompt therapy. And I know not every speech therapist does it, but they would literally take her mouth and move it. And for her, she was not able to speak because she did not have the coordination to move her mouth properly. So I think them taking her mouth and moving it in the right way really helped her. And the other thing that I think for Olive, and I don't know if you guys find this, whenever we like take a trip, or even if we're in our neighborhood and do something different, that's like an experience, I find she kind of grows. Like I find a lot of growth from her when we put her out of her everyday environment. So I would just recommend that when you can.
Thank you. I just want to talk about education in school a little bit, past, present, current. For us, Lily was in the public school system starting at 3, and then last year we pulled her out. We didn't want to pull her out. We really wanted her to be a part of the community, especially as an only child. We wanted her to be around typical peers. It was just not right for her. It was a really hard decision. She's now in a very, very specialized school with 6 classrooms, and there's 4 kids in her class. My anxiety level has decreased about 80%. And we just really feel like she's being taken care of. To your point about her speech device, it's there 24/7. At the public school, she'd have to walk really far to this only bathroom she was allowed to use. And sometimes she would have accidents just because, it took so long to get there. So we had been really apprehensive about having such a self-contained situation, but I think it's the best fit for her right now.
We're actually very lucky where we are when it comes to schooling. So Lena was able to attend, a special ed preschool that focused on ABA, which seemed to really help us when she was little. And then as she transitioned into, kindergarten, public schooling near us had some phenomenal programs. Our local school district has, I think it's 5 or 6 different levels of special ed schooling. Hosted at different elementary schools, and she's in one. It's called the MOVE program. Again, she was not walking at all, not even close to it when she started that. So it factors a lot of movement and everything like that into the learning, into the day. Some of the kids in her class are still not walking, but there's a lot of that goes along with it. She's been in the same class with teacher from kindergarten now through 6th grade. So now my anxiety is peaking because next year she has to go into a new program. So we'll see there. Luckily, though, we've had such an open relationship with the school, with her teacher, who is super invested in her, who, treats her as one of her own kids, but probably pushes her a lot harder than we do. So we've been very lucky there. There are lots of programs out there. We've gone through it with our son. You know, one program doesn't fit. You know better than anybody else. Okay, so when you're going through that, when you're doing those dreaded IEPs, when you start doing that stuff, fight. You know better than anybody else out there. Make sure that you're standing up for what you need. If you have to get support, get extra support in there. But there are enough programs out there. There are enough good things out there that you don't have to settle for something that's not going to help your kid. So just make sure you are that amazing advocate and that you're revisiting it constantly. And there is light. Like, you can get to that. You can find the right program that's going to help your kid thrive.
Where we live in a town about 23,000. There is one school district. There's a handful of private schools, but none of them have any special ed programs. So I don't feel like we had a whole lot of choice, besides moving. But we are happy where we've been. We've had, Emma's had the same teacher since kindergarten. She's in 4th grade now. So we've only got one more year before she has to switch programs, and that will be a huge change for her because same kind of deal. She loves her teacher. Her teacher loves her. We see her them out in the community, and she's always so excited to see her teacher. Good, bad, or indifferent. My biggest thing has always been that she's happy to go to school because I feel like she will learn if she's there. If she doesn't like school or she's not happy being there, there's nothing to help the learning along. I don't know if she goes there more to socialize or, but she's always happy to be there, happy to go. It's never been a struggle or anything getting her to go in the doors. Along the same lines, I think if you do have to change programs like Liz was talking about, and you find out it's not the right fit, don't be scared to change your mind again and go back. It's hard to know exactly what something's going to be until you try it, your child's tried it for a while, and then you decide, well, this wasn't great. I need to regroup and try something different. Because with our kids, we can't just know what the next step is and know that they're going to fit in right there. That's going to be the spot for them. So sometimes we have to regroup and go backwards and try something that we already tried or try it differently. I think that's a lot of the time if we're having an issue, I kind of have to try to think outside the box, try to think of new ways to do stuff that I hadn't had a thought of before. I also just wanted to say, I know I said my daughter's extremely verbal, and she is. It was not on a, quote, normal trajectory. I looked at some of the notes because she's 10 now, so we kind of forget. But when she was around 3 years old, she had 30 words, and we used sign language a lot too at that point. I think when she was like 4.5, she had about 70 words verbalized that we could count. Within the next year, we stopped counting because we couldn't. We were talking about reading a little bit. She can sight read maybe 102 words is what her teacher did the other day with her. And it's just, her memory is incredible. She doesn't forget a thing or an event. And like you were talking about going to events, there was a time that anytime we went on vacation, we'd come back and even the teacher started noticing, like, oh my gosh, what happens when she goes with you guys? I'm like, I don't know. It's just the new things. It helps them grow and experience new things. So, but I'm a little worried for middle school for her. Honestly, we don't know if what we're gonna do, if we're gonna try to change paths or do something different. But throwing her into the middle school is hard enough for any child at that age. But we're a little nervous about that ourselves and still unsure what our plan is. So.
I'm happy to say we are on the other side of the middle school transition, and all is well. I think we go into any transition with optimism and skepticism, right? And so go back to early 2016, and Emmeline had just come out of early intervention and was going into our local public preschool program, and we just weren't super on board with. We just didn't have those great feelings that she was in the right place. That was our first DDX3X conference. She had just turned 3, and we learned about intensive clinic-based ABA therapy. We went back home. We got an autism diagnosis from our developmental pediatrician, and that allowed insurance to kick in to be able to have 3 years of intensive clinic-based ABA therapy. Emmeline started walking during that time. She started signing. It was magical for her. I mean, she went from a kid who'd never interacted with people, who really did fit all the autism criteria, to a kid who was just as social as she could be. And so we went to the transition to kindergarten with that same kind of optimism, but skepticism. And she flourished in those 6 years in elementary school. And we had incredible teachers who just kept believing in her and impressing us. And so when we went to make that transition recently to the middle school, it was again that, but these people love her. We can't take her away. They don't know her. And it's been wonderful. Again, I go back to Liz's comment about, if your anxiety is telling you this is not the right place, by all means, look elsewhere. We kind of went in with that, like, are we going to have to do something different? And thankfully, it's been so far so good. And so have hope.
It really is. Annabelle went to our public school, our neighborhood school, until she was 10. And at that point, she was in a self-contained class. And at that point, we were told we were going to be sent out of district. So we started looking for a school. And she started at an ABA school. That seems to be a trend here, which was huge for her. They embraced her talker. She had a lot of structure. She had an extended school day. And she was there until this year. And she started at a residential school in January, and that's going great.
Olive has sort of always gone to a private school. We live in the city, and I think the public schools can either be great or not great. So we were lucky enough to get her into a private school that is ending this year. So I'm starting the process of looking for new schools for her. But she's a very eager child and loves to learn. So I feel like she's always been really excited about whatever school she's at, which has made me feel the same. But it's been great for her. She's been at the same school, like I said, for 5-6 years. And it's just been a great experience. We really haven't had any issues with it.
One more question, topic, and then we'll open it up to the audience. Coping skills. Everyone's going to be on the spot to talk about their coping skills. Besides wine and gummies. It is not, yes, besides wine and gummies. We all know it is not easy at all. Got connected to this woman who lives in Shanghai the other day, and to figure out if she should move to our town. And I said, it depends, like how social, how important is being social to you? And she's like, all my friends are special needs parents. Those are the only people who get it, which I thought was really interesting, because I feel that way about Alex. He's the only person who really, really, really gets it, because he's living through it with me day by day. Sometimes it's really hard to be around families who have typical kids. We don't have one. And when they complain that their child didn't get on the baseball team, you're like, really? And then I was kind of having a hard week. So I'm like, I'm going to find a support group. And there happened to be one that weekend, and I went, and someone's like, my kid has ADHD. And I'm like, you do not belong here. So everybody handles things differently. So I'm just curious, especially the dad perspective.
All right. So I'll give you a couple things that have helped me. One, my daily thing, some kind of something you do for yourself. Okay. So it's hard. We're so in that mode of, I got to take care of my kids. They need extra care, extra this. I have to be supportive. My wife and I have to be a team. We have to go after everything together. Like you got to find something you do. So for me, it's running. So like I go for a run every single morning, and it's how I structure my day. And I bleed the stress off from the previous day, and I turn my brain off for a few minutes, right? Because now I'm not worried about work. I'm not worried about the kids. I'm not worried about anything else at that moment. So I got a little bit of time. That's just me. So that's where I do it. So that, I swear, that's the only thing that's kept me slightly sane for the last 11 years. Two, make your connections in the special need community, right? And the special needs community is great, but like Liz said, that's a huge range. Okay. So these kinds of events, talk to people, because you talk to somebody. I remember, I don't want to go on forever, but I remember walking into the Chicago DDX3X meeting in 2016, I think it was, terrified. So we had gotten our diagnosis the December before. We didn't know what to do. We still didn't know what it was. No doctor could tell us anything. There was some study out there with 25 kids in it from the Netherlands, I think, or something like that. That was all we knew. We showed up and Kate had already connected with people on Facebook, so she felt better. At that point, I still was anti-social media. I wasn't doing anything. I signed up for Facebook right afterwards because of this group. But she had started connecting with a few people, and we walked into the lobby of the hotel. Still scared, not knowing what's going on. The elevator opens, right? And it's Tiff from San Diego with Lucy, who has the exact same birthday as my daughter, and connection across. And it was like, nope, we found our people. And since then, we've always said, like, this is our tribe. This is like my healing weekend every year when we get to do this, or Arkansas or something. So we do everything we can to try to be there. But when you can connect with people who 100% know what you're going through, even though these kids present differently, right? And there's a range of skills and abilities and everything. There's a whole bunch of stuff that we all have in common. One, we are all teaching our doctors everything because they've never heard of it. I haven't yet spoke to any kind of medical professional that's ever even heard of it before seeing us write down DDX3X. Two, you can almost always tell a DDX3X kid, okay? There is no like, oh, they have this forehead or they have this eye set or anything, but you can look, and we do it all the time. We'll be out like, hey, I wonder if, is that a DDX3X kid? Like, and part of it's them, part of it's the joy, and there is this magnetism that they have that just attracts people to them, which is so cool. So, find that group, people you can talk to where there is zero judgment, 100% understanding, and you can vent out your darkest thoughts because they've all had them too. And then the last thing I will say is just, and this is probably the hardest thing, but it's kind of like resetting your expectations, right? Because we all grieve that life that we wanted for our kids. And we know they're not going to have exactly whatever that ideal was in our head, but they're going to have an amazing life, and we're going to make sure they have it, and we're going to celebrate like crazy. Like I said, I have videos on my phone from Lena at like 1.5 years because I was timing her sitting unassisted for 1.5 minutes. And it was like, oh my God, she didn't fall over yet. And then in the video, you watch her fall, and I dropped the phone trying to catch her, right? It doesn't matter. I sent that to so many people who were so excited. And I guarantee you, there's no typical kid out there where we were so excited that they sat for 1.5 minutes that we were timing it and sending it to people. So blow up those wins, love those wins, share them with everybody, and you will have people cheering these kids on like you've never seen. And that's how you find your wins. So that's how I cope. That's how I'm surviving right now.
I guess for me, when I know when I'm getting overwhelmed, sometimes I feel like it's Groundhog Day. You just wake up and it's like the same thing. Okay, whatever, the same mess, same stuff every day. And you kind of feel like you're, losing your mind a little bit. You're like, I'm pretty sure I just did this the day before and the day before. Just even verbalizing it to another adult in your life. There are people that, besides my husband, and he works a lot too. And so sometimes during deer season, when he's working a lot, it does feel like, like I'm on my own. Now Avery is older, she's 14. So we are able to leave her at the house with Emma. Emma thinks she's old enough to be at the house alone by herself now, but she had mozzarella in the toaster a couple days ago. So, and I was at the house. Literally, you don't know what's going to happen when you turn your back. So finding people in your life that they may not know specifically everything your child's going with, but if you need a couple hours, there are, find people that you can trust, whether it's, a friend from school or their teacher. Their teacher might even be willing to watch your kid for a couple hours if you need just a mental break. But I think sometimes just verbalizing it, like, I just feel overwhelmed today. I think we all go up and down on our emotions and workload, and then you got your kids on top of it. And so some days are just more than others. And you don't know why you're feeling the extra stress that day, but recognizing it and seeing what you can do to help bring that down, what the things are that you like to do that help reduce your stress. because I'd say for me, I used to, or I feel like I used to have a decently clean house and not messes everywhere. And I liked it that way. And, you know, it'd be great to have that now, but I have to let go of things that I can't control sometimes. And Emma really likes to pour things out of anything. I can't even describe all the things that's happened to, or like you said about Apple Care, I've lost count of how many times they've replaced our iPad. We had a bent chassis one time. Did anybody know that iPads have chassis? I was told that when I went to Best Buy, that you got a bent chassis, and I'm like, wait, what?
Sorry, a what?
A bent chassis,
Hey,
like a
Vanya needs
car?
on mute.
Yeah, it's like the metal frame of your iPad. And the screen wasn't broken, but we had a bent chassis. Anyways, there's all kinds of random things that come up that you never thought you would deal with. 12, 15 iPads, I don't know. They've been in the bathtub. I can't even. Exactly. I don't know why they're not waterproof. Anyways, but sometimes you just laugh at some of the things that have gone on because you never could have thought that was going to happen, like the mozzarella in the toaster. I don't even have explanations for some of the things that happen, but they're funny stories later on. But I guess, yeah, find the joy in the funny things too, because you just never know what's
Hey,
going to happen.
Vanya needs on mute. Vanya needs to mute.
Yes.
I would say for our family, we have surrounded ourselves with a lot of sitters and helpers and grandparents and aunts, because I think our caregiving bandwidth is low some days, and having some folks who can step in and help. We are in Virginia, where we are very fortunate to have, at present moment, a Medicaid waiver that helps us with that. And so that has been, I think, even just having someone come in and help on Saturdays so that Patrick can play golf, I can do some shopping or something with other girls, or he can, yeah, so that he can do something with the other girls, is really, for us, I think, how we recharge our caregiver battery, and show up as our best selves, for Emmeline.
I would definitely agree with all of this. It's definitely the other parents. I have a couple of groups that will go to dinner, or we'll just sit and talk and laugh and tell these stories that no one else understands, but you immediately understand if you've dealt with a child like this. So just connect with as many special needs parents as you can.
I agree with everything everybody said. I think, and there's nights that like I'll notice my husband's at his tipping point, and I'll just be like, I will deal with this. And he does the same for me. I sometimes feel like one parent and 2 kids is just as hard as 2 parents and 2 kids. So sometimes it's better to like give your spouse a break, and I don't know, it helps us to take turns sometimes. And I know this really also sounds weird, but I feel like getting involved in the foundation, because I know we're all here to want to find treatments, but at least I feel like I'm being proactive at a good goal. So I feel like that can help sometimes too.
I was going to say that. I thought it'd be too soft for me.
So one other just quick thing I want to throw out there, just piggybacking on a couple of the other, we talk about that extra support, caregiver support, respite. Find out the programs that are out there. Some of them are hard to find. Kate did, I don't know how much research and how many years of filling out forms and everything. But like New York has something called self-direction. And I know there's other states out there that have things. So, I mean, you may have seen a young girl, Maddie, who's running around with our kids today. So she's a respite worker. She works in our house 4 days a week, taking care of Lena, helping her out, taking her out into the community. We set her budget, we set her pay, but it's paid for by a budget out of New York State. Like, there are amazing things out there, but no one's just volunteering it to you. So you have to ask questions and you have to find out what's out there, because you need a break, and you're going to need a break, and you need someone you can trust, and you need people that are invested. And we're now on, I think, our 3rd main respite person, because unfortunately we've had people move on. It's life-changing, and the people who no longer work with Lena still keep track of her, and they're still asking about her, and they're still invested in her because that connection they make. But Don't try to do it all on your own. And sometimes, you can rely on family. Family always wants to help. It's not quite the same. So make sure you are finding out. That's where other special needs groups in your area can help. Just keep asking those questions because there are programs out there, and anything you can get, take it. Don't ever feel bad if it's like, oh, they're going to give me a budget. I don't want charity. I don't care. Okay. You go take whatever you can get because it's there for you. Take advantage of it. And use everything you can get. But again, I don't know if there's a good way to go online and just find out what's available because it seems like that stuff tends to be hidden. Ask a lot of questions and don't be satisfied with no, there's nothing. Okay, just keep asking because it is out there and it can be amazing.
Does anyone have any questions in the audience for any parents? Nothing? Yes, great.
Hi, thank you, parents. Your bravery for being up there and sharing it's heartwarming. And I get to speak from that. My daughter is outside of school and now has embarked on a whole new place in terms of the school bus does stop coming. And that's sobering, but also take everything that you are experiencing in the districts and the schools and the communities you're establishing, because those are the ones that are going to get you to a new school bus, a new journey, a new piece. My question for you is, yes, self-direction, anything that's out there to help our kids. And also another comment on physicians and providers, hold their hands to the fire. They're in areas of, of the country that they should be expected to be questioned and queried to know more, do better for our kids. And, the piece of recognizing that nothing happens until it happens, I think you alluded to. So my daughter as well was fine with some of the things, and now new fine things are starting to show, which never showed before. So that fresh review all the time and always being interested in your kids and your families and creating positive environments is really, really critical. And these are the building blocks. So thank you.
Any comments? Great. You guys pass this.
I guess we'll give the 40-year-old perspective, but that was amazing. I mean, you guys are doing it. I just want to reinforce the dairy thing that you said. So when Lindsay was 5, we took her off dairy, and it wasn't lactose; it was the protein in dairy, and potty training happened, and words came. So really look at those food allergies and see. And get trained in the special ed law. Every state in our country has a parent training and information center. I used to run the one for Massachusetts. Learn your rights. You're not in the school districts to make friends. You're there to make sure your child has what they need as they go through. The friendship stuff isn't important, but getting involved is. So Jack joined the school board. I ran parent groups. Now we sit on committees in the adult world. Lindsay's on the self-direction advisory board with me for Massachusetts. Give back, because then you know who the players are. So before 22, it's what you know. After 22, it's who you know. And I can't reinforce that enough. Separate schools are important to learn those skills and grow, but if you can keep their life full and meaningful outside of school or in school, Lindsay is active in our community because we wanted her to be with typical peers. And she wasn't fully included, but she had a one-to-one aide, and she was in that school and knowing people, and they knew her. And now as a 40-year-old, they're her coworkers. They're the people working out with her at the Y. They remember her from school. So as much as you can, because after 22, life gets quiet. And it's harder to keep it full and meaningful. And that's what we spend our days doing with her self-direction program. Yeah, so, and the part about what you do for yourself, one thing that I think really helped me was going to therapy. So give yourself the gift of therapy, especially if you can find a therapist that understands raising a child with a disability. Because see, I was going to cure her until she was, and until today, I didn't know that that was possible. And when I got to therapy, it wasn't her that needed curing, it was me. I had to get rid of the thought that she needed to be cured because she wasn't broken. She's amazing, and she's given me a career and us a full life. So it's so cool to be here and hear your thoughts and travel back in time, and know that you have technology that you can get broken. We didn't have that, right? But they're speaking and they're using those communication devices. I think that's fabulous. And what's next, right? So let's get excited about all the things that are coming. So our life is wide open, and because I have all that training, if I can help you as you travel through transitions or IEPs, I'm on the Facebook group and I'm happy to help. So good luck.
First of all, I just want to thank you so much for all the parents who've shared their experiences. It really means a lot to those of us, including me, who is just very new at this diagnosis and have like very little experience. I guess I just had a question, like a very specific question for those of you guys who deal with constipation. When did it start and how do you deal with it?
Did all the things, they all my phone.
Anyone else? Questions? Yeah. Yeah, just quickly. You're mentioning noise, and I don't know, get out there. Probably that. I was wondering. It might be worth this trip. How do you treat it? How's it treated?
Give us something. Regret so much.
I'm just curious, I didn't see a lot of that, that you have failure.
We've had that. We still have that.
And then still, so we finally have one. But now we're trying to see this her tripling now. Again, she's not technically classified as failure to thrive, but we're like wrong. The file. I mean, I think. Yeah, we had, she was growing, but.
How old is your daughter? How old is she?
Even she was fine. She did really great, but as far as maybe, like, it's just really, it's hard to
Yeah.
get used to the gameplay.
So do you know Kate's farm? It's, yeah, we've got, I mean, our daughter's 12 and she's tiny, tiny. And it's like, they have a non-dairy version, but it's like chocolate and it's like 800 calories and we give it to her before bed. We're like, oh, 800 calories. So highly recommend it. Kate's farm, yeah. Yes. Yeah, and we just order like a ton at a time. Yeah, sucks them down. Oh, well, it's been a journey because she was on ADHD meds, so she stopped eating and we just took her off of them. So she's now, we're trying to get her to gain the weight back, but she did. She initially gained like 8 pounds. Yeah.
And she's on a special diet now. Trying to count calories in, but it's literally that she's not
Yeah.
She's home all day.
And then conversely, if Beth was here, she would say that there's also a contingency of girls that gain weight once they hit puberty and weight gain becomes an issue.
Fourth grade, I mean, that's impractical for them all the time. But she doesn't eat. And like, it's just establishing. But she took a little bit of the trial, like 4 or 5 years, and all of a sudden she shot up and gained weight, and she's like in 75, 80, I checked in. And I'm sure she's taller than my older daughter. But just one day, I don't know, she started eating nonstop. Yeah, anything sensory. I don't know, she's still not there. It's like almost oral sensation, but it's nonstop. So I didn't bring it out. We went full container, like big old full container. He's awesome. My 6 feet got one thing, and then. Like. Yeah. Yeah.
Food in front of her. And it's just, I don't understand, I don't even sense her just put ketchup all over the hair, like, it's so funny, right? It's, yeah, it's just using utensils, yeah. But that's helpful, but like, you know, things that she will enjoy, I'm like, 4 calories without blueberries.
Yeah.
They're great, but I'm like, just. And I, you know, weight gain is very, very instrumental in so many other health factors.
I remember being one of the pediatricians was telling me I had butter and everything. I like that. Yeah.
We haven't had like another 5,000. Or worry. environment like this, and she will. We try to every short sheets and things like that, just to make sure you get a good job. So it's easier to stop that down. Those pouches with all the vegetables, fruits, and everything like that, and all species. We never travel anywhere without each. Those will. And all of the. You butter it with them. I know you can bang on. Other times she will, others, but. Never gotten to the point. Scary health concerns, but it goes on and we stress about it constantly. We just got travel shakes and things like that. That just helps. Always having some on hand. And trying not to stress too much about it. We will get very. On speed. You need more than she can. You know, okay, sometimes that's when you find it off. Well, you know, she can't tell us that her stomach hurts, so she can't tell us that she's things like that. Oh, God. So just keep trying to listen, harm that kind of stuff. Find something, you know. Anymore.
Yeah.
But it's just that was all go to. And sometime after that. But I don't need to start that now. She. The area. Wondering how these I.
I just wanted to add to that. My daughter was also in the first percentile classified failure to thrive. Under 10% is what we were told for failure to thrive. And then when you get down to the first percentile, you know, this is a first world country. Like you feel like your child is starving and you don't understand how they are surviving. But we use Neocate, and that comes with a variety of flavors. They also have a junior and then more calories for as your kid gets older. Our daughter was on Neocate up until she's about 3. And then after that, her chronic vomiting and diarrhea went away. She was vomiting and had diarrhea every day. So just hang in there and just pack in the calories. Don't worry about it. That's all.
Some and
Anyone else? Thanks, everybody. What time is it? Quarter to what? Quarter to 4. Sorry, checking the schedule. I think we have a break. Quarter to 4. Oh. It is now. Yeah, it's. Yeah.
Great, that started an hour ago.
I'm Kate. I got my turn. Yeah.
I grew up on Long Island. My parents are out. My mom was out in Suffolk County, so. There you go. Okay. Right.
So it's a really, it's a long, and it involves a lot of stuff. But I'm actually there. I'm starting it my son. Also, sorry. So we're, yeah. But I want to get it. I have, she's on the meter. And I know people are very upset that I don't want to do it. I'm bad, right? And I swear, I'm trying to get to know you. Personally, I think that needs to be here.
Oh, I don't think they really, I don't need to do that.
And just be able to do this, but not much. So I need to get through the steps, yeah.
Yeah, so it's a long process, but what's great is once you get it, that's the value, right, carries into adult. So it's like a budget that gets issued, that can be used, or maybe you can take advantage of most of it when they're young, because they're in school, when they're getting colleges. But if it's like, they help cover, or at least give money towards like hand, different programs that are outside of school, like respite, digital care, reimbursement for family respite and things too, so like family members helping care for them, obviously they're not charging you, but you do something for them, you can get some money back for expenses there. We've done grant, my trip there. Yeah, part of your budget, you get issues, a certain amount, they go each thing. So then you get hooked up to different organizations and you hire somebody for that organization. Did you meet Maddie? Yeah, Maddie's amazing, right? So Maddie takes, she'll take me out and take me, but hey, you take her to party, shopping, smoking that, she'll take out, she's over every day, she's there to help him be a muscle. So she's there to party, she stays like 5 days, she'll do, she's obviously taking good care of us too, so you're paying her at the same time, and it's not us paying, like. But, and there's different levels, like there's respite, and there's something that's combat, which we got her into the community, you know, and that's where you're saying, you know, like that, not like, nothing, and she can actually make some money doing it, but your whole budget goes through, and it's that, and it's all these other programs you can allocate it to, and you have a care manager, you have this team that helps you manage, you're constantly going to reassess what money you have available, how it's allocated every year, and it's really locked in now, it follows them into their 20s. So they're still getting money available in budget issue, the school is not following things anymore. That's it's so critical to get in with that. Plus, helps you. You can go and be like, I need to get out of here. And I say, no, it's taking care of you. And you can have maybe you're hiring in. Like, yeah, you can that's an average. There's a lot to it, so and we're starting to get the information on it, but it was one of those things we were like, oh God, this sounds too good to be true. It's like this whole process. I don't know. It can be challenging to find it because when you get to hire, so you work with an organization and they might help connect you with people, but they don't just send, it's not like they send somebody like, oh, like this person, you're interviewing them and you're hiring. They're just paid through that agency. We actually have like, where we convinced her to go get certified by the agency if we wanted her to be on a rental partnership, and who did it, and now we're. So like you can involve, you have our last person, Maddie, who was a friend of mine for years and years and years, who had been a preschool teacher in the past, who did a first teacher in the past, and everything like that. But the kids anyway wanted to do it, so like 2 years. So it's amazing. It really, really is. See if I'm still back.
She's amazing, but.
And you can have one. You just have, you can have somebody in the background that they do 2 days here and there. Or me, yes, it doesn't matter. They can sit down. Like I said, you'll be blowing your hair out going through the process. It takes time. There's a lot to it. It kills me when I talk to people who are crazy like that. I don't know how you're doing it. It's, you gotta find people. Look at that, and like I said, I'm 8 minutes, all of the time again, it's getting to the first time. We'll send you that, but I should be able to see it, so I can see it. Presenting people with disabilities, things like that, it's something like that, it really is part of is involved in also. So, yeah, check some of that stuff out. It's weird because the information's out there. It doesn't really make sense. Like I didn't understand it, and we go through it. Like, wait, they just give us like, oh, it's a budget issue that we can use or we can use. But we work through it again. So nice to be able to have a beer. Yeah. You help find somebody that you can find. So then you got time. Just like, we don't have to worry about the being received with that, or they're going to go take on for bite, you're going to go outside and play, whatever it is. No, she's safe. Somebody's watching her, and somebody's invested in her. And it doesn't have to be me. It's so nice to do that and not feel bad about it. Yeah, check it out. And then we'll definitely like let us know when it. Yeah. Okay. Let's really read you. But you know, it should be out, yeah, and listen, it's one of those too, when you need to talk through something, like reach out, and that's, Kate does a lot of that, because she's on the leadership team, like, reach and everything, she during the day still and everything like that, we're happy to help things move a lot. How old is your daughter? 8 months. Yeah, I assume you're doing all like the stuff like that. Yeah. Okay. Oh, nice. Yeah, that was, man, all that stuff. It was so crazy. So, and we have, so we knew, like, you know, born that they were watching their notes, so we didn't know what to go back. Okay, it's awesome, it's not right. So what? Like, you need to get therapy. And then, so we also made the program for Sunday. And that's the other thing too. Unfortunately, very once you like those things that you were going to, are you sure? All right, but it's one of those things, like we've had opportunities to like think definitely one of the better ones. And they go, and there's a lot of good students down there. So You have years before you can get to that. So, yeah, no, it's nice to meet you. Are you? What exactly is happening?
Microphone. Where do you want your water?
Are you trying? Yes, yes, we are. I love it, yes. And then how do you get? But after there, you go second. There, you're not doing that. You know, maybe have that. Oh, okay, yeah, we're going so think about it. He's used to volunteering. That's exactly what the parent conference. I was like, I was in the parent panel, but I thought maybe I asked if they wanted to do it. And I was like, oh, click on it. So I like Wi-Fi classroom right now. Yeah.
Like walking with you or?
No, you're fine, thank you. So, I know, it's weird, but everyone's like, I find that big. Yeah. Yes, that's so like, yeah.
He said walk. And that's got.
All right, I'll be right back. Yeah.
So can they give us maybe a walk?
Yeah. Yeah, well, it's so funny. Yeah, yeah. And like. No, it's a big one. I have Yeah, so, you know, like, I tried one of those right now. I think
Hi, everyone. We are going to, hello. Hello. Hi, sorry. So we're gonna take about 5 more minutes of people just, you know, taking a break, networking. And then we will go ahead and do the closing remarks. And then you're welcome to either leave, go back to your small groups, whatever you want to do. So just 5 minutes and then we'll finish up formally. And we'll talk more about it later, but we will have dinner this evening at 6:30 at Roost and everyone is invited.
Yeah, we don't let, so we were, and my brother wants that. And then how, what I'm doing, but I, then I'll say, I don't, like, when I, like, being out, yeah, and really, right? Big sense, right? So I'm gonna have it. That's funny, I'll be in the back. Yeah, basically. And it should be maybe. Yeah. They're working on getting back. Properly. Yes.
Oh, testing. Okay. If everyone could take a seat, please. Almost done, I promise. Test, test, test. Hello. Where's Nick Tambone? Paging Nick Tambone. Thank you. Hi, everyone. Just a couple of things. Thank you. I know it's been a long day, so thank you, everybody. Just want to go over our funding priorities. We talked about a lot of things today, a lot of science that's happening, but science requires money and a lot of it. So our first priority is to continue funding the natural history studies, our registry, and our centers of excellence. The centers of excellence have providers who are experts in DDX3X. So a lot of people talked about how their doctors don't know anything, but if you go see Dr. Caceres in Chicago, she knows everything. And we want to do more of them. We want to have them around the country. So there are go-to places. Oh, I just gave you a shout out. Our goal is for there to be go-to places across the country, so people receive the best care by the most knowledgeable doctors. And those also cost the foundation money. The hospitals need funding for those to take place. We also heard a lot of exciting news about gene therapy and the prospects there. For this to continue from today, basically on, we need $400,000 to support the gene therapy research next year. And then after that, we're going to need an additional $200,000 to continue to fund our natural history study, registry, and centers of excellence. So the $400,000 for gene therapy is just for next year. And I think Dr. Gray really illustrated it's a very long-term project that's going to require significant funding. Next slide. So now more than ever, it's imperative that we come together to raise money for those affected. And we are working really hard with our teams to fund research, to find treatments, and we need our population and our community's support at whatever level is possible for you. We have a toolkit and graphics. If anybody has any questions about how to run a fundraiser, we have experts. Chelsea's really knowledgeable. You can talk to the Grace family. You can talk to Sarah Golden, who did a lemonade stand, and there's definitely creative ways of raising money, including pajamas. So we are super excited. There is a company that came to us. Their company is called Sleepy Saturday, and they make amazing soft pajamas that are usually in 5-star hotels. And they're doing a deal with us where they are going to make them for us with DDX3X logos. They're going to charge us cost, and then they are available for everybody to buy, and all of the proceeds go directly to the foundation. We have to hurry because this all happened very quickly. So the deadline to get them in time for the holidays is October 26th. Highly recommend for teachers, therapists, anybody in your child's life. And we have a few designs. Those are the kiddo designs. Next slide, Chelsea. And then there are adult sizes, kid sizes. For adults, there's long and shorts. Same with the kids. The kids ones have the logos, and they will be on our website and also on Beth's store's website, Kirna Sabate, as well. So kirnasabate.com or DDX3X, you can place your pajama orders, tell your friends, tell grandparents. It's a really nice, easy way to raise money for the foundation. So if we sell 4,000 pajamas, we'll get $400,000 for gene therapy. So that is our goal, 4,000 pajamas. And that it. And then also, we're thrilled to partner with Double Good Popcorn. I don't know all the details, but we will keep 50% of all the sales. It's going to start October 27th at 9:46. Interesting. And end October 31st. You can download it on the Double Good app. There's all the information. We'll make sure this is all. Hi. Who's wait? Yeah.
I was gonna intro. We are doing the Nourish design, the DDX mandala. We're gonna be doing that pre-order again before
Great.
the holidays.
Great.
So.
I thought you knew about the popcorn. Okay.
A little background on the popcorn. Candy Sorrentino, who is a member of the Leadership Advisory Board, she's not here today, unfortunately. But one of her child's schools, PTA, did this, and they raised like $28,000. So the way it works, I mean, they're obviously much larger than us, but we could do it. So you get a 4-day window. We're going to do it leading up to Halloween, like trick or treat yourself. And it's just a super easy, as Liz said, we'll have all of this information added on Facebook, and we'll send it out via e-blast. And you can just share it with your family and friends. All they have to do is buy the popcorn, and we get 50% of the sales.
Great. Next slide. Also, when you send your pajamas, you can also send minted holiday cards. If you use this code, you'll get 20% off, and then 15% of the sales will go to the foundation. So you just put in the promo code fundraise DDX3X. If you want to get the video that was shown today and post it on your social media or send it to all your friends and family, asking them to buy pajamas, this is the code. So if everybody wants to grab a picture of that to send it to everybody, we want it to go viral. That is the goal. Next slide. Upcoming dates. So these are our big fundraisers for the year. Giving Tuesday, which is the Tuesday after Thanksgiving. Rare Disease Day this next year is going to be February 28th. There's going to be a European conference in Italy next year, and more details coming on that. We don't yet know the date or the possibilities in terms of next year's conference. But if we do have one, there's going to be a mandatory pajama day. So we need everybody to go and buy their pajamas to support the conference. And that's it. Thank you all so much for being here. I hope everybody found it really rewarding. I know that I did. If anybody has any questions, please come up and talk to Chelsea or I and Beth at dinner tonight. And I hope to see everybody later. Thank you.